parte de mi investigación postdoctoral y lo que dio base a este tema que he estado trabajando. Si tienen preguntas, en cualquier momento, por favor, párenme. I'm going to be easy. If you guys have any questions throughout the talk, please do stop me. I hope you don't mind. I'd like to walk around, so I'm not going to stand at the lectern. And without further ado, let me go on. First of all, I would like to thank everyone that has made this work possible. We recently moved from the Wayne State University, which is in Detroit, down to the University of North Texas. All the work that I'm going to talk about today was done at Wayne State, so I have to acknowledge them, and also funding from NIH, the National Institutes of Health. The computing time is gratefully acknowledged by Wayne State and Silicon Mechanics. And the people that actually did the work that I'm going to talk about today were Rebecca Sweat, who did all the SNP work, Dong Fang, who did all the mechanistic work I'm going to talk about on ALK-D, and more recently Alice Walker and Pavel Silverstruck, who've done, in my view, an amazing job for this SNP characterization that I'm going to tell you about. So, someone, a dear friend and colleague who's now at McMaster once told me that it seems that I have research attention deficit disorder, research ADD, because in my group we like to do a lot of different things. We do a lot of methods development related to quantum mechanics and the combination of quantum mechanics with classical mechanics, which we call QMMM. We're very interested in the applications of computational chemistry to investigate enzymes that are related to DNA replication and repair, which is the topic of today's talk. And the work from here actually was an offshoot which inspired this cancer biomarker determination. And so this is, I'm going to tell you the story of how going from these blobs, which represent electron density and non-bonded interactions, which we'll hear a lot more later on today, led to this, and also some other applications that we've been doing. Okay, so just to give you an idea of what I'm going to talk about today, I'll tell you about cancer biomarkers, why they're important, what are single nucleotide polymorphisms, I'm just going to call them SNPs from now on, and also genome-wide association studies, or GWAS, and how we can use the information garnered from these studies to find these cancer biomarkers. And now the trick is how we use this information and how we link the enzymes and the mutations that come from these SNPs to different diseases. In the case we're interested in is cancer. In particular, we look at DNA repair. So we've looked at several different enzyme families. Today I'm going to talk about DNA polymerases a little bit, and then a lot more focused on ALK-D family enzymes. So I'll tell you about the reaction mechanism of ALK-D and the insights that we've gotten through computational simulations on these, and then go on to the most recent work, how we've been able to find a SNP on ALK-DH7 that is related to prostate cancer, and what this mutation actually does. So before I start, just wanted to give you an idea of how inspiration, at least for me, comes about. I'm sorry, the resolution is not that great. But this is a woven sculpture that is in the National Art Gallery in Washington, D.C. It's called the Renwick Gallery. And the piece is called 1.8 millionths of a second. And does anyone have any idea what this piece represents? I can give you a clue. It has to do with something that we use in computational chemistry. It's a representation of the map of the energy that was released during the Tohoku earthquake and tsunami. So it's basically an energy surface. It's a representation of an energy surface, okay? As quantum chemists, that's how I started. I consider myself a quantum chemist first and foremost. This is one of the things that is at the base of our understanding, right? Born-Oppenheimer theory begets the potential energy surfaces. I was very fortunate when I was in Wayne State University to count as one of my mentors and great friends and colleagues, Professor Schlegel, who is a world expert on potential energy surfaces, so many thanks to him for this image. So basically, when we're doing computational chemistry, if we want to study, for example, reactions, reaction mechanisms, et cetera, what we have to do is we have to explore potential energy surfaces, okay? So when I was a postdoc at the National Institutes of Health, I was lucky enough to work not only with a great computational chemist, but also with very many outstanding biochemists, protein crystallographers, microbiologists. And so I learned a lot of biology. And what a lot of the group at NIH in that particular institute were interested in was DNA repair, and in particular DNA polymerases and how DNA polymerases aid in repair. So one of the things that I did was I studied the reaction mechanism of this little machine over here. This is called DNA polymerase lambda. And what DNA polymerase lambda does is if there's a break, a double-stranded or single-stranded break on somewhere on the DNA, this initiates a cascade of different, there's two main cascades that are that can be activated. One is called base excision repair. The other one is non-homologous end joining. So DNA polymerase lambda will fill the gap in DNA. It generally fills small gaps, 1 to 2 nucleotides long. So what we did was investigate using computers how the reaction to put in the correct nucleotide into the gap of the DNA actually worked. So why is this important? Well, it turns out that DNA gets damaged relatively easily, and there's many, many different factors. So just by different agents that are caused by metabolism or viral infection, chemical exposure, the typical radiation, you go out without sunblock, UV exposure, this will cause double-stranded breaks, etc. When the DNA gets damaged, this will elicit a host of different responses. For example, transcriptional activation, apoptosis, which is something that we don't want, or DNA repair. And so that's the topic of my talk. So obviously, if DNA gets repaired, then we're done, right? So it turns out you can go to Amazon and buy this DNA repair cream, and then we're done. So now you can ask, why do we want to study DNA repair? Well, I wish it was this easy, right? So one of the things that happened when I was working as a postdoc and doing this investigation of the DNA polymerase lambda is, in addition to finding out how the reaction, this is a representation of the active site, there's 2 metals in the active site, and what happens is this is the triphosphate of the incoming nucleotide, and what happens is a bond between this oxygen and this phosphorus gets formed, and obviously the enzyme catalyzes this reaction. But it's not only the metals that act to catalyze the reaction, you have the whole scaffold, right? You have the whole enzyme. So there's a lot of different residues that are important for this reaction. We were able to predict several of these residues, all the ones that are in red, we predicted from our calculations. And 2 to 3 years later, our collaborators at NIH actually mutated E529, lysine 472, arginine 386, lysine 422. And indeed, I was very happy and very relieved also that our predictions were actually correct, and they do, in fact, impact catalysis. So what happens when you mutate any of these residues is, depending on which residue, the enzyme loses activity, so it doesn't catalyze the reaction as efficiently. It's slower, or, which is not good, or a bigger problem is, for example, if you mess with this lysine 472, you make the enzyme not be as faithful. What does that mean? It starts adding incorrect nucleotides. So what that means is you start getting mutations. And this is not good, because as we know, or as you remember from biochemistry, we have 3 billion. In English, 3 billion is 10 to the 9. In Spanish, just so that we're all on the same page, in Spanish is 10 to the 12, right? So a billion in Spanish is a million million. In English, it means 1,000 million. So 3 billion is 3,000 billion, 3,000 million. But anyway, so when you have mutations in regions of the DNA that actually code for proteins, this can become a big problem. So after this, and learning all this microbiology and structural biochemistry at NIH, I started to wonder, well, could there be mutations on these residues that don't kill the enzyme but make it do things that it shouldn't? And are these conserved evolutionarily in other polymerases? It turns out that DNA polymerases are very numerous. There are very many different polymerases. There's 18 human polymerases, and then from there, for all of the other organisms, there's a huge amount of polymerases. So one of the things I did was try to figure out if any of these residues are conserved. Lo and behold, of the 9 residues that we predicted, there's 5 that are at least partially, if not totally conserved. This was not an easy exercise because doing the alignment of the polymerases is not easy. Some polymerases are very small, with around 100 amino acids. Others are huge, like 3,000. So it was an interesting experiment. But this brought about another question. And this is where this idea of how a quantum chemist got interested in cancer research. So what I started wondering was, if you have a mutation on those, obviously those residues are evolutionarily important. So what happens if you have a single nucleotide polymorphism? So the hypothesis that I came up with was, if you have an individual with a SNP on a DNA polymerase gene that has an impact on the function of the protein that is related to cancer, then this might be used as a biomarker. This could have a predisposition towards cancer for those people that carry that mutation. And now my next step was, well, what are you going to do about it, right? So what we did was, since we're computational people, we developed an algorithm in the associated software to search for SNPs on single genes of interest and determine the functional impact. So use our computational tools. Once we know where the mutations are, use our computers to figure out what these mutations do, if anything. Okay, so the traditional way of doing this, so if you're coming from systems biology, biostatistics, and the biology side, the traditional view of genome-wide association studies is you have what's called a cohort, so a bunch of people that have a particular disease, let's say, for example, prostate cancer. And then you gather a whole host, hopefully of the same number or around the same number, of non-patients, so what we would call the healthy group, and you do a gene sequencing project. So you sequence the entire genome of every single person in that group. So you have, let's say, 2,000 patients, 2,000 controls, and then you have all these SNPs. Remember that 3 billion bases for all of them. So you do the genotyping, and then you compare. But you're comparing 3 billion bases for 2,000 patients and 3 billion bases for each of the 2,000 controls, right? And from there, you have to do a lot of statistics and very stringent statistics to figure out if a mutation that happens here does not happen here, and does that mean anything, okay? That's what a lot of biostatisticians do, among other things. But the problem is you're looking at the entire genome, okay? And the reason why people do this is because obviously in this way you're not introducing a bias. You're not saying, okay, I think it's because these enzymes are over here, this is my pet enzyme, or my pet protein, or my pet, or I'm interested in this particular metabolic pathway, so I'm just going to look there. So this is more unbiased. But since we're computational chemists and we're interested in DNA repair, and we know that DNA repair is directly, or there's a huge link between DNA repair and cancer. That's why the 2015 Nobel Prize in Chemistry was awarded. Then what we decided to do was, well, why don't we use some of the information we already have? So let's see what SNPs can actually do. If you guys remember, the central dogma of biology is you have DNA, you translate it to RNA, and then you transcribe it to proteins, right? That's really easy in prokaryotes, but in eukaryotes we have what's called alternative splicing. So if you have a mutation on the DNA, you can have 3 different outcomes. So if you have a mutation of the DNA in a site that's called an intron, then because of alternative splicing, that's going to get cut out, and it's not going to affect the final protein. It may not affect it. I'm not going to get into details. If you have a mutation on an exon, depending on what that mutation is, because of the codon code, you can have 2 possibilities. One is it's a synonymous mutation. So basically, in the codon that gets mutated codes for the exact same protein amino acid in that position. So in the end, the final protein has the exact same sequence as before. Or you can have what's called a non-synonymous exonic SNP. In that case, the mutation actually does change the amino acid. And so you have a SNP that changes the final protein, okay? And those are the ones that we're interested in right now. So what we decided to do is use this software and the algorithm that we came up with at this comparison stage, and then look at significance and make predictions. So we select our gene of interest. In this case, I'm going to talk a little bit about the DNA polymerases because that's the first example that we looked at. We generate the gene or exon. You can do a genomic or exomic database in a searchable form, search for the SNPs, do statistics, and then try to figure out if the mutations are in a piece of the structure that has been crystallized. So to give you an idea of the size of the problem, if we have 12 different cancer studies, that's what we started with, that was 7.4 million SNPs. We use our phenotype algorithm, we called it hidden SNPs, for hypothesis-driven SNP search. This reduced from 7.4 million to 2,500 SNPs. And then from these, how many are exonic? 534. And then we do the statistical analysis. And this is just a snapshot of the stuff that we found. So we found 79 statistically significant SNPs for 4 different cancer phenotypes for DNA polymerases. So the phenotypes are lung, breast, prostate, and melanoma. All of these SNPs have been previously overlooked. So the people that did the original studies using statistics had overlooked these SNPs because once we have only 2,000 SNPs, we can actually move the statistical threshold for what we call significance, the p-value, by just a hair. And with that, we're actually able to come up with this. Another thing that we can do is we can do what we, as computational chemists, we would call correlation analysis. How, if you have 2 mutations, are they synergistic? So we can do haplotypes for 2 or 3 SNPs, and we did indeed find in my pet enzyme Pol lambda a haplotype for 2 SNPs that gives an increase in risk for breast cancer, and another one in Pol gamma that gives an increase for prostate cancer. So that was all very exciting, and we've been collaborating with some cancer researchers in the Karmanos Cancer Institute in Detroit to try to follow this up, especially the Pol gamma haplotype. So that's work in progress. I'm not going to talk more about that. What I would like to change gears a little bit and come more to the computational chemistry part and then show you how from this we can actually get all the way to cancer and how computational chemistry can help us in understanding this at a very granular detail. So like I said, there's a lot of different DNA damages that can happen. There's a lot of different DNA repair enzymes. One of the ones that we're interested in is this ALK-B in the human family of enzymes. ALK-B and the human homologs are in charge of dealkylating damaged nucleobases. They're part of what this superfamily that's called the iron and alpha-ketoglutarate dependent enzymes. So the reason why they're called this is because they have a non-heme iron in the active site and alpha-ketoglutarate as a cofactor. And using these 2 things in the active site, they catalyze an oxidative reaction in the active site. They can act on a huge array of different substrates, single-stranded DNA, double-stranded DNA, RNA, and even histones. All of these enzymes have a conserved active site motif. There's 2 histidines and an aspartate that coordinate the iron in the active site. Why is DNA alkylation important? Or, well, why don't we want alkylation on DNA? It's a very common type of damage to DNA. Also, if you bend my arm, there's DNA methylation is also important, for example, for DNA expression, yeah, exactly, for epigenetic marking. But that's a very specific 5-methyl cytosine, right? So, most of the other alkylation of DNA is deleterious, meaning that it damages DNA, it causes bad things to happen. And there's many different ways that this can happen by chemicals from outside the cell or the body or internal. They can cause mutations or cytotoxicity, and obviously because of this, cells have developed a lot of ways to fix this. You can fix it by means of what's called the base excision repair pathway, where you chop off a little bit of DNA where this damage is, and then you patch it up, for example, using DNA Pol lambda, or you can do it directly. The enzymes that do this dealkylation directly are the ALK-BH family enzymes. So when I was an undergrad working here on my undergrad thesis with Miguel Castro, my thesis topic was nickel clusters and investigating a lot of spin properties of nickel clusters. So I was always very interested in metals and also in spin arrangements in different metals. So when I found out about this enzyme, I got very interested because it has iron, right? And iron is a partially filled D shell atom, in this case iron 2. So this is the proposed reaction mechanism from experimental studies and also based on the tidal enzyme of the superfamily taurin D. And basically, I'm not going to go through the whole thing, but you start with iron 2 and then have molecular oxygen and this cofactor alpha ketoglutarate, oxidize iron all the way to iron 4 to form this what's called this ferryl intermediate iron 4 oxo. And in the process, alpha ketoglutarate was oxidized to succinate and CO2 was released. So this is the first phase of the reaction, is the activation of the dioxygen. Once you have this ferryl, obviously this is a very reactive intermediate, and what's going to happen is this oxygen will abstract the hydrogen, in this case from the methyl that's damaging this adenine, to form a hydroxyl, an iron 3 hydroxyl. And this hydroxyl, the next step in the reaction is called a rebound. The OH comes back to this free radical. This is a very labile and unstable intermediate that will disproportionate to form formaldehyde in the free base. And then you regenerate iron 2 and you can go for another cycle. So what we're interested in is the second phase. The first thing that we looked at was this step right here, which is the rate limiting step in the reaction, this hydrogen abstraction. So the question is, since we have iron, and in this case iron 4 oxo, what is the arrangement of the spins of all the electrons? Well, if you have iron 4 oxo, you can have a quintet or a triplet, and then we can calculate the energies. But it turns out, depending on the ligands that you have around your iron, the oxygen can transfer an electron to the iron, and depending on which spin and which orbital that electron occupies, you can have an antiferromagnetically coupled iron 3 oxyl or a ferromagnetically coupled iron 3 oxyl. So I'll call this in high spin iron and intermediate spin iron. Okay? So one of the first questions that we wanted to answer is what's going on with this iron and oxygen in the enzyme active site, because this is going to have huge implications for catalysis. So how do we do this computationally, right? If you're trying to find the reaction of an enzyme, you have to do the calculation. An enzyme is, what have you, 5,000, 10,000 atoms. This one is not too big, it's around 7,000 atoms, but then you have to put it in water so that you're simulating some stuff correctly, and then you add counter ions and a bunch of other stuff. In the end, you can end up with something between 50,000 to 100,000 atoms. For those of you that are computational chemists, you know that simulating 100,000 atoms with quantum mechanics is impossible right now. So Professor Karplus, who unfortunately couldn't join us, but he was going to be here, was one of the originators of this QMMM method. The idea is you have classical mechanics where you can simulate big systems like enzymes. You have quantum mechanics where you have access to electronic structure and reactivity. And if you combine both, for example, you just simulate the enzyme active site with a quantum mechanical program and then everything else classically, then you can look at a bunch of different things. And this was the reason for the 2013 Nobel Prize to Warshel, Levitt, and Professor Karplus. People have used it for a whole host of different things, not only mechanisms, substrate binding, and a bunch of other things. This review is obviously a little bit dated, but there's been a huge increase in the use of this particular methodology to investigate not only enzymes, but now also zeolites, metal organic frameworks, and a bunch of other things. Okay? So that was our method of choice. I'm going to go relatively fast on the methods. I'll be happy to answer questions if you have any. But basically, what we have is the first coordination shell of the iron and a couple other things. Obviously, the adenine, and this is the reaction that we're going to investigate is the transfer of this hydrogen, right? So we have around 45,000 atoms. We use DFT, and 61GDP. I'll be happy to talk more about that DFT and obviously the spin and all that stuff. There's always questions about that. We try to be very careful. So the first thing that we have to do is get the crystal structure from our experimental colleagues and then run some classical mechanics to make sure everything's okay. And then the trick is, how do you explore this potential energy surface? There's many ways to try to figure out how you can connect the reactant and the product. In this case, our reactant is this structure right here. The product will be with the hydrogen already transferred to the oxygen. Our method of choice is what we call a quadratic string method, where we can basically optimize the entire path in one shot, and then we can optimize the transition state. Once we have this, this is the potential energy surface. But obviously, when you have enzymes and water at 300 Kelvin, you have motions, so you have enthalpy, you have entropy. So what we really want to look at is the free energy, the Gibbs free energy, and this is what we can compare to experiment. So we do a technique called free energy derivation. And then one more thing, if we want to analyze the wave function, one way to do it would be to just look at the orbitals, and you can do canonical orbitals or localized orbitals or a bunch of other host of things. But looking at that for QMM, well, at least for me, even for clusters, was always kind of tedious. There's other ways to do this. I'm going to show you this conceptual computational chemistry way using the electron localization function, ELF, and non-covalent interaction, or NCI. And NCI, we're fortunate to have Julia Contreras here. She's actually the developer of NCI, so I'm not going to talk too much about it. I'm going to let her tell you about it. I think that's part of your talk. Okay, so this is our optimized reactant. As you can see, the iron here in pink is in an octahedral field with 4 equatorial ligands, 1 histidine, 1 aspartate, the succinate, and 1 water, and 2 axial ligands, the histidine and the oxo, right here. And this is the hydrogen that's going to get abstracted. Okay? So the reaction profile, that was the first thing that we looked at. And what we had predicted is you can have the iron 4 oxo or you can have iron 3 oxyl either in a quintet or a triplet, right? Septet we also investigated, but septet is way too high in energy, so I'm not going to talk about it at all. So what I'm showing here, I know it's a lot of data, but basically these numbers over here are the molecule spin densities on selected atoms. So in the iron, the oxygen, the hydrogen that is getting transferred, and the carbon. Black and red are the quintets. Blue and purple are the triplets. And the only thing that I want to highlight from this profile is this particular thing here. So we start, according to our calculations, in the intermediate spin quintet state, and then at some point during the reaction, we have what's called an intersystem crossover. So we go from the intermediate spin state wave function to the high spin wave function, and that allows the reaction to happen. Now the question is this for real, or is this just an artifact of our calculations, right? And we're using DFT, so we can talk a lot about that. But hopefully we'll convince you that we're not too far off. So we had to characterize the minimum energy crossing point. I don't have too much time to get into the details. I'll be happy to talk more about that. I know there's a lot of people that are experts in this field. We should be using multi-reference and all that stuff. We've done some tests. This seems to pan out. I'll give you a little bit of validation on that. But basically, we started the reactant state for the look at the dotted line. That's the free energy. The solid line, that's the free energy surface. We go to the crossover, go to the high spin state, get to the transition state with a barrier around 18.6 kcals, and then drop down to that second intermediate. Our barrier of 18.6 kcals per mole is relatively close to the experimental estimate. The experimental estimate is around 20 kcals per mole. So whenever we're doing calculations with these kinds of huge approximations and we're within 2 kcals per mole, in the QMM community, we basically just start jumping up and down because we basically think, okay, this is a representative path of the collection of reaction paths. Okay, so I'd be remiss if I didn't show something like this. We spent a lot of time doing this rendering. What I'm going to show you is a rendering of the reaction mechanism. The DNA base is right over here. This is the methyl that's damaging the DNA base, and this is the hydrogen underneath this gray surface that's going to get abstracted. So this is a combination of these two methods that I was telling you about that will allow us to analyze the wave function and where the electrons are. So the gray surfaces are the electron localization function results, and they represent where the electron pairs are. So if you think about like Lewis structures with the electron pairs, you know where the bonds are. So this represents the bond between the carbon and the hydrogen and the electrons that form part of that electron bond. Okay? The green surfaces, blue surfaces, and red surfaces are the non-covalent interactions, so NCI surfaces. And they represent, for example, the hydrogen bond. So there's a hydrogen bond between this water and this oxygen over here. And also we can see the coordination shell where there's not quite a bond. Coordination bonds are very strong, but not quite covalent bonds. But you can actually see that there's kind of like a donut here in the NCI surface. So NCI also allows us to investigate coordination bonds, which I think is really fascinating. So let me see if I can get this to work. What you will see is this water moves out of the way. We get a stretching of the oxygen-iron distance. This polarizes the oxygen. And then you hit the transition state, which is a shared bond between these three atoms, iron, hydrogen, and carbon. The hydrogen always transfers with the with the electron. And then it drops down to form a very strong hydrogen bond with this aspartate over here, which stabilizes the intermediate at a lower energy than the reactant, and thus we get an exergic reaction. One more thing that we can look at is this minimum energy crossing point that I was talking about. So by definition, this is the point in the Born-Oppenheimer surface where the structure has the exact same orientation of the nuclei, but the wave functions are different. Okay? It's a degenerate state in the hypersurface. And when you look at it with the molecular orbitals at the molecular orbital level, it's relatively difficult. Well, to me, it's difficult to see. But when we look at it with these ELF and NCI analysis, things to me became really a lot easier to understand. So if you compare these two figures, this is the intermediate spin, this is where you start in the reactant, this is the high spin, this surface you have to jump to go to the transition state. If you compare the two images, or if you subtract them, you basically see that they're almost exactly the same, except for the surface on this oxygen. So in the intermediate spin, you have two what are called basins, and we can actually integrate the electron density in these basins, and we can calculate how many electrons we have in each of these basins. We can do a multipolar expansion and then figure out dipoles and quadrupoles, et cetera, et cetera. When we do that, we see that the dipoles over here are very small. When we do that same analysis over here, we actually see that we have the same number of electrons when we sum these two to this surface over here, but there's a huge dipole that is actually pointing right at the hydrogen. So we posited that you need to traverse through this minimum energy crossing point to the high spin surface to activate the oxygen in order to abstract the hydrogen. So another thing that we can do is this same prediction of the residues around the active site. I'm not going to spend too much time because I'm running out of time. But we predicted a bunch. We have some agreement with experiment with the same trends. We don't expect to have the exact same agreement. That would be basically impossible. We don't get the same agreement over here. We think we know why. And if you have questions about that, I'll be happy to answer those. I'm going to go fast on this, but this is basically the rest of that second phase of the reaction. What happens once you have that hydroxyl activation? Does it really do rebound? And this goes more to the biology of the reaction, because it turns out that ALK-B and most of the ADH enzymes like a basic pH between 7.5 and 8. And also there's another piece of information from experiment that was really interesting to us. This is from a collaborator, Bob Hausinger, who was crucial for some of the results I'm going to show you later, in a moment. He's been working on this tidal enzyme taurin D, which catalyzes the removal of a sulfide from taurin using the same motif, this iron 4 oxo. So this enzyme was the first one that was studied for the whole superfamily, and this is where the whole idea of this OH rebound started. But recently, well, in 2010, they did time-resolved Raman spectroscopy, and they found a new peak. And so they proposed these two possibilities for the reaction, where you can assign this new peak to a structure where you have the hydroxyl rebounded all the way to the carbon, or where the oxygen actually is has two bonds shared between the iron and the carbon. In their paper, what they were predicting was more leaning towards this. So now the question is, this is on the second phase of the reaction, so what's going to happen there? Since the reaction happens in a basic in a basic environment, we decided, okay, let's do a couple of tests. Let's run a bunch of calculations to see what's the reaction with water in this equatorial position, or replace it with a hydroxyl. I'm not going to explain all these, but these are all the possible steps either considered or stepwise reactions going for the reaction with a water in that equatorial position, or with a hydroxyl in that equatorial position. My first graduate student, Dong Fang, did an outstanding job doing all these calculations. I'm just going to show you the potential energy profiles. And just to point out that if we have a water in that equatorial position, the barrier is just way too high. But if we have the hydroxyl, the reaction after forming that second intermediate is really easy. And this was something that was not predicted from just structure, because from a crystallography standpoint, you see the oxygen around the iron coordination shell, but you don't know if it's a hydroxyl or a water or what, because you can't see the hydrogens, right? So from our calculations, we were able to propose an alternative pathway. So let me just quickly summarize this whole reactivity. The minimum energy path for the rate-limiting step, that second phase, involves a crossing between two different surfaces. We found 9 different residues that destabilize the transition state, and they're conserved with other human enzymes in the same family. The reactant for the rate-limiting step involves this intermediate spin iron 3 oxyl, and this is part of what I was saying of the backup of those claims. There's been a bunch of other calculations on AlkB since we published this, and a little bit before. And not only that, but also on these metal organic frameworks that have the exact, it really interestingly have the same motif, an iron and an oxygen, and they catalyze the abstraction of a hydrogen from alkanes. And they did multi-reference calculations on those, and they do see the exact same number of electrons as I presented in that reaction profile. So I thought that was really cool. And finally, from our calculations, we predicted an alternative pathway with a hydroxyl, which may explain the basic optimal pH for the repair catalyzed by AlkB. Okay, so I just gave you a bunch of really technical computational chemistry and all these nitty-gritty details about the potential energy surface and where the electrons are and all that stuff. But if I go to my colleagues from the NIH, they would say, so what? What does that matter to biology, right? So now what we're trying to do is bring this more to the real applications, right? So we do a lot of basic research, but now what's the importance? So what we did was apply our SNP search algorithm, hidden SNPs, to all this, to find if there's any SNPs on AlkB enzymes, the human enzymes. There's 9 enzymes in this family, ABH, AlkBH 1 through 8, and FTO. We found we found a couple of thousand actually. Out of those 2,000, there's only a handful that are actually statistically significant. So that means that they are more prevalent in cancer patients than in non-cancer patients. And out of all those, there's only one exonic non-synonymous SNP. It's this RS7540, and it appears on AlkBH7. And what this mutation does is that it transforms an arginine 191, to a glin. And now the next question that we had to ask was, and we had to answer, does this matter? Does this actually affect the protein? So the first thing is look at the structure. Where is the mutation? Is it in part of the structure that is crystallized? Because sometimes when you do crystallography, you have to chop off different parts of the protein. In this case, we were really lucky. The mutation is actually part of the protein that is crystallized. The arginine 191 is this residue right here. This is just to give you an idea of the location with respect to the active site. The distance between the center of mass of this arginine and the iron is around 22 angstroms. So it's pretty far away, right? But we know there's a loss rhythm and a bunch of other things. So now the question is, if you mess with this arginine, does that have any impact? So how does RS7540 affect AlkBH7? And for this, to answer this question, we turn to classical mechanics. So we perform MD simulations on the wild-type enzyme and on the mutant with both the co-substrate, alpha Kg, and also with the product, with succinate, just to see if something changes before or after the reaction has happened in the active site. We ran a bunch of dynamics, so in total 1.5 microseconds for each system, using Amber 14 and a bunch of other things. I'll be happy to talk more about the technicalities there. Also looked at NCI to see hydrogen bonds and all that stuff in the active site and at the mutation site. Professor Adrian Reutberg is present. He's done amazing work. We're really happy to use his script to calculate cofactor and co-substrate binding, because we found something that I think is really interesting. Hopefully you'll find it interesting too. And then from these predictions, we actually contacted Bob Hausinger, who was down the road at Michigan State, is a very dear friend and collaborator, and he actually did experiments to see if the predictions that we made were panned out, if they were correct. So what were those predictions? So basically, after we ran dynamics, I hope you guys can see it in the back, what happened was, what I'm showing here is in gray the wild type structure that has the arginine, which is the wild type enzyme, which forms a hydrogen bond to this D182, this aspartate 182. In the mutant, after 500 nanoseconds, with the glin, this distance increases dramatically. And what that does is it slightly messes up with all these beta sheets. This is called a jelly roll fold, and that in turn changes the active site. So when we saw that the active site changed, this is what brought us to, well, what does that do to the binding? So let me show you a little bit of the structure changes. So in the wild type enzyme, the solid lines down here are the distance between the histidine and the iron. As you can see, in 500 nanoseconds, nothing happens. I'll be right. Thank you. The active site is solid as a rock. In the mutant, all hell breaks loose, basically. The distances are much higher, and also these two lines up here represent the hydrogen bond between that arginine and that aspartate. As you can see in the wild type enzyme, it fluctuates, but it's around 6 angstroms. In the mutant, that hydrogen bond disappears. Okay? Also, this affects the dynamics. This is a difference correlation plot. So basically how every atom is correlated, the motion of each atom is correlated to each other atom. And as you can see, not all of the atoms get affected by this mutation, but here's the chain of the dynamic correlation that affects the active site, that goes from the mutation site through the jelly roll fold to the active site. And I'm going to show this movie a couple of times just to give you guys an idea of what happens. This is a movie of right before the enzyme actually changes conformation from the wild type to the mutant. So what I'm showing in these 3 panels is the full enzyme. So here we have the mutant enzyme in the conformation that is corresponding to the wild type. So you will see this guy move out of the way, and then everything gets messed up. And then this is a close-up of the active site and of the mutation site. So as you can see here, when you have the mutation, the glin does make a hydrogen bond. There's a tiny little blue surface there, but it's very weak. As you will see, as the movie progresses, this hydrogen bond will disappear, and this glin will move out of the way. And in the active site, the iron will just go everywhere. Okay? So here we go. We have the hydrogen bond, and then all of a sudden, with the thermal fluctuations, the hydrogen bond breaks, and then all hell breaks loose. Let me try to do it again, just to talk about these NCI surfaces. You can see the hydrogen bond there, and then it breaks, and then the oxygen breaks the coordination bonds with the histidines, and then it gains a bunch of other new coordination bonds with waters and a bunch of other aspartates. Okay? So obviously, if you have this iron moving in the active site and you lose that coordination, what happens to the binding of the co-substrate and the cofactor? Well, in the wild type, the binding, this is just the enthalpic component, because the entropic, we didn't even want to mess with it. But this is just to give us a qualitative idea. And this is just to show you that the binding doesn't change, right? The numbers in this case are very qualitative, and I would argue irrelevant. What's interesting is the change in binding. As you can see here, in the wild type, this co-substrate and the iron remain bound. In the mutant, they unbind. So that was our prediction. We went to Bob, and he did this beautiful UV-vis experiment in an anaerobic conditions where they measure the absorbance of the iron oxo, this ferrule intermediate in the wild type and the R191Q variant, and they take the difference with and without the iron. And as you can see, in the wild type, you have a very nice defined peak. In the variant, there's nothing. So we were basically we were in the plasmid, and I know this is negative, and it's because of the subtraction. So what they did, this is all in vitro. I requested the plasmid. They did all the hard work of expressing the enzyme and doing the experiments. But what we believe that this is a nice validation of our prediction. So just to summarize, we have this new method called hidden SNPs to search for the C SNPs. It doesn't have to be cancer on any protein. We did QMM simulations that revealed intersystem crossing in an alternative mechanism for the second phase of the reaction. We found a new SNP that is statistically significantly correlated to prostate cancer on ALK-BH7. And not only that, we were able to predict, using computers, what the effect of that SNP or that mutant, the product of that SNP is, and confirm it experimentally. We've also used hidden SNPs on a bunch of other DNA proteins, DNA repair proteins, and for a whole host of different phenotypes, and we're collaborating with several cancer researchers, so stay tuned. Again, I would like to thank my group. As I said, we were in Detroit, and in Detroit, I had a little piece of home, which was very nice. Literally a block away from my office, we had the Detroit Institute of Art, which has the Diego Rivera Detroit Industry Mural. So the joke in the group is that the Cisneros group likes to work hard, think hard, and play hard. And with that, gracias.
Thank you much, Andres. We would like to take some questions from the audience. Please, we have these colleagues who can take the microphone to you. Please.
Thank you very much for a very great talk. It's great to see former students doing this really great job over there. And the question has to do with the intersystem crossing step. Mm-hmm. And I wonder if it matters the relative timescales for the system to be exploring the configuration in which the intersystem crossing would occur.
Oh.
And the timescale of the step itself, which is not quite finite, I guess.
Yes, that's a great question. So I'm going to punt on that because, so the answer is yes, it definitely matters. And obviously, since this is a dynamic system, so what I didn't have time to explain is that what enables this intersystem crossing is, among many other things, but the main thing that enables this intersystem crossing is the increase in the distance between the iron and the oxygen. And it's basically an interplay between exchange repulsion and orbital energy. And that's what allows the electron to hop from either the paired in the intermediate spin state, being paired in one orbital, this is the sigma star orbital basically, to the pi star orbital. So when you stretch the iron-oxygen bond, the energy of the pi orbital comes down, and this is what happens. And this is similar to this thing, this moth was also observed and where they did multi-reference. But obviously, when you have thermal fluctuations, you have this thing going on. So I would love to be able to simulate something like that, but it's extremely hard. So there are people, I just came back from this conference on QMMM, where they are actually doing dynamics with QMMM, where they propagate not only the classical degrees of freedom, but also the quantum. So that would be a possibility, but it's obviously extremely difficult and extremely time-consuming. But it's a great question, yes.
Thank you.
I saw one hand over there. Yes?
Great talk, congratulations.
Thank you.
Any plans on doing NMR and a further refinement on the mutated enzyme, just to follow dynamics?
Oh, yeah, well, I mean, this is literally just the first step, and the people that we got the plasmid from, it's these Chinese group that did the first, well, the only crystal structure. So they're protein crystallographers, and we're hopefully about to get comments on the review of the paper. So hopefully this will elicit more, so literally these are fresh off the computer results, not even published, but hopefully published soon. But yeah, I would love people to do something like that.
Here. So this mutation to arginine to glutamine, it's affecting the enzyme activity, and if so, how much?
So they didn't do enzyme activity yet because those are they're even more difficult than the binding assays, and they only it's one of the rate limiting factors, as you guys well know, especially for experimental, is the money, right? So how much money you have to express and all these things. So for the absorbance, for the binding, they didn't have to express as much protein. That's my understanding, at least. So one of the ideas that we have is hopefully apply for a grant to get more money so that we can do stuff like that, because that would be fantastic.
Take another question there.
Miguel.
Well, I am really proud of you.
Gracias, Miguel.
Why occur those oxidation states on iron 3 and 4? There are some experiments?
Yes. So they've done Mossbauer, EPR, and Raman, and they actually have trapped some of the intermediates going from iron 2 when they don't have, like in anaerobic conditions with no alpha-ketoglutarate. They know it's iron 2, and then they've trapped that ferrule intermediate and characterized it with Mossbauer, not only on ALK-B but also on taurine and then a bunch of other enzymes. So that is well characterized. They know that it goes through those. However, the whole path, you can't trap every intermediate, right? So that's where the computers come in.
And also the chosen functional is the appropriate one.
I was expecting that question. Thank you. Yes. So we did a lot of work on that because obviously DFT, right? And it's a single reference, single determinant, single determinant wave function and single determinant method. So what we did was the way we benchmarked it was we looked at 7 different functionals and 3 different basis sets combinations. We looked at the entire path, reactant, transition state, and product. And we looked at concordance with known experimental facts. Like we know that the reactant has to be a quintet state. We know that the barrier has to be a certain height. And we know that the product has to be also a quintet state. So of all the functionals that we tried, the only one that satisfied all of these different criteria was omega B97XD and 631 GDP.
Okay, thank you for the very nice talk. Perhaps I misunderstood. When you talk about the ELF and the NCI analysis, I understood that the transfer of the hydrogen atom, it was in the form of a neutral atom, right?
It was what?
It was in the form of a neutral atom. It comes with each electron.
Yes, it's a hydrogen atom transfer.
Okay.
Yes. Yeah.
And then you mentioned that it was important the OH minus anion.
Yes.
And then my question is the reaction going with free radicals or is it ionic?
It's that's a great question. So take this with a grain of salt, right? Because the analysis is it's coming from topological analysis of a DFT wave function, right? So this is a qualitative analysis. Let me see if I can go back to some. Oh, I hit it right on the. Wasn't meaning to do that, but let me see here maybe. Basically, when we have the hydrogen halfway through in the transition state, since we have that structure, we can actually do the ELF and the integration and all that stuff. So we do know that it's a basin that is shared between the carbon and the oxygen from this DFT wave function with 2 electrons in that basin. As the reaction progresses and you go to the product, one of the electrons appears to remain on this carbon, and the other one oxidizes the iron. It goes all the way to the iron, and you form that hydrogen bond. So, sorry, reduces. Yeah, I'm going to get my title revoked if I say oxidation without hydrogen transfer, right? Anyway, so this is consistent with the Mossbauer experiments where they know that this carbon here will have, it will be a free radical. So we think it's a free radical reaction, but yeah.
Okay, thank you.
We would like to take one last question from a student.
Yes,
for a change.
please.
We can try to convince your teachers that they give you a point or something.
I would like to ask, sorry, about your hypothesis. It was that SNPs were related with cancer.
Yes.
But it was not clear for me. Do you find if they are really related? Or
Yes. So let me see if I can go here. I went a little bit too far, sorry. And now, okay, I was just trying to get back to the list. So basically, once we get this list, the idea is we pick a particular family, so in this case, for example, ALK-B. And what we do is, sorry I wasn't clear about this, I was trying to rush a little bit and I rushed too much. The data where we get these SNPs from, it's called the database of genotypes and phenotypes, and the database of SNPs. This is an NIH database, and it comes from all of the studies, well, all of the NIH-funded studies that have been done on cancer. There's literally now dozens of cancer studies and hundreds of just all sorts of different disease studies. And each of these studies has between 2,000 and 5,000 patients and 2,000 and 5,000 controls. So what we do is we just look from the data of these databases. In this case, for prostate cancer, it was 2,000 patients. We just look at the part of the genome that codes for these ALK-B enzymes and search for SNPs, okay, for all of the SNPs just on the patients. And then we have to do statistics. We have to see the SNPs that we found, and like I said, we found 2,000 SNPs, right, on all of the ALK-B enzymes. Do any of these 2,000 SNPs appear only on the patients compared to the non-patients, to the healthy cohort? And out of those 2,000, only this list over here is statistically significant. So that means that these mutations, these SNPs, appear prevalently only on patients that have prostate cancer compared to non-patients. Granted, this is not all the patients, obviously, and cancer is a multi-gene disease, so not single one. But here, since we're interested in going beyond and trying to explain the molecular level effects of some of these mutations, that's why we're looking at these.
Okay, one final. So the relationship is because of malfunctioning of the enzymes, or which is the...
Yes. So now, well, that's the question. And that's a great question, and that gets right to the bottom of the issue, right? So this is what cancer researchers call either a driver mutation. So does the cancer arise because of this mutation, or does the mutation arise afterward? Once you got prostate cancer, you get this mutation. And so that's the next question that we have to answer. So now we have an idea of, yes, there's a correlation between the cancer, the particular cancer, and this mutation. Does it cause it? Correlation sometimes does not imply causation, right?
Yes.
Great question. Okay. Thank you.
Thank you.
We will have a very last question from the head of the department, so I couldn't say no.
Thanks. No, I was interested in this bit you said, and the question is pertinent in that sense. What is first? But then this mutation may have arisen by a failure of Paul Lambda working.
Yes, for example.
Paul Lambda, if I am not wrong, is not a replicative enzyme.
No.
It's a repair enzyme, and actually a recombination enzyme.
Yes.
And therefore, you miss in recombination, then you mutate an enzyme that has to correct the mistakes, and then that's some sort of a snowball.
Exactly.
And you start accumulating mutations.
Yes.
Therefore, cancer is just a product of accumulation of mutations.
Yes.
That's a very interesting thing of all this.
Absolutely. And it may not only be Paul Lambda, but a whole host of others, right? And then you get into like systems biology, which pathways connect to all of these, because there's many very different pathways for DNA repair and recombination and all of those. So yeah, that's fantastic hypothesis. Thank you.
Please thank Andres Cisneros for a great
Thank you.
conference.
Thank you much.
I'm giving you this memento. You will remember this celebration.
I'll remember this. Thank you.
I want to remind every one of you that tomorrow we will have some time devoted to students who want to talk with our guests for this symposium. They will be available so you can talk to them, ask questions maybe personally that you didn't want to ask now. Okay, so wait for that tomorrow. Okay, we'll change for our second conference in this symposium. We will have Juan de Pablo, and I will ask him again to talk a little bit about this school and his experiences with it. Thank you much.
All right. Va a estar difícil. It is a pleasure to be here. Es un gusto estar aquí otra vez. En los últimos 2 o 3 años he dado varias charlas en este auditorio. Y tengo que confesar, cada vez que hablo en este auditorio se me pone la carne de gallina un poquito. No por la emoción, pero porque los exámenes finales eran siempre. Y soy como los perros de Pavlov, empiezo a ponerme nervioso cuando estoy en este auditorio. So thank you so much for inviting me. On the way here in the taxi, the cab driver was complaining. He was saying that the city is a mess. It rains, there's traffic, there's pollution, everything sucks. And then I was thinking, you know what? It can always be a little bit worse. This is a picture of the University of Chicago on a beautiful day, on a normal day. It looks a little bit more like that. And I was telling the cab driver that the recent study found that the air in Chicago is 75% bullets. And the study, if you read closely, was endorsed by famous atmospheric scientist John Molina. Not Mario Molina, but John Molina. All right, let me thank my collaborators on this work. The late John Whedon, Dan Schwartz, and Juan Fernandez. And these are really the students and postdocs that have done all of this work, especially John Chin, who's now at Stanford, Ray Zhang, and Josh LeCue. All right, so what we're trying to do in our work is really understand how it is that DNA can actually go from being a fiber that is just 2 nanometers wide, but 2 meters long, into a collapsed structure that actually measures just tens of microns or just a couple of microns in some cases. The way this is done in nature is by having DNA wrap around these histone proteins, and this collapses into a fiber that collapses again and again and again until you have your chromosome. This process really remains a mystery. We know more or less what happens here. We know a little bit about what happens here, but then everything else is still an unknown. And yet, the three-dimensional is this on, or is this better? And yet, the three-dimensional structure of this chromosome is really, this is probably worse. is what governs all of genetics. So our efforts really are trying to understand how you go from here to here. Now, to put things in perspective, it is useful to remember that the characteristic length scale for a DNA turn is 2 nanometers and 3 nanometers here. And yet, when you collapse a long strand of DNA into a virus, for example, you actually package about 300,000 nanometers into a capsule that measures only 50 nanometers across. To put things in perspective, that is equivalent to taking about 2 kilometers of rebar, folding it, and packaging it all into a Volkswagen Beetle. What a virus does, day in and day out, is comparable to what I'm showing you here. And the way that nature does it in a virus is by applying very subtle forces that twist and turn the DNA to funnel it into a virus. Of course, in industry, we would do it using these giant machines that measure about 3 stories high. In nature, you have these tiny little motor proteins that actually do that very, very elegantly without making any mistakes. Part of what we do in our work is to try to come up with models of DNA that allow us to describe these processes quantitatively so that we can one day understand how chromatin folds and how it behaves. So what I'm doing what I will do today is tell you then about how DNA starts to behave at micrometer and millimeter scales, how we go to nanometer scales, and eventually how we go to angstroms. Now this is a talk that is really in reverse from what you normally do in quantum chemistry. You start from electrons and atoms, and then you try to go all the way up. One of the premises of my presentation is that there are some problems that are so complicated and for which we have so much available data that it's actually worth it to do everything at the same time and use whatever information you can take from any source to get to the answers that we're looking for. All right, so the first part of this talk is really motivated by work in which we're trying to design optical mapping methods that will allow us to sequence DNA. We already saw a very nice introduction in the previous lecture about why it is important to understand where everything is in DNA. One of the methods that we've been involved with to try to sequence DNA is shown here. So what you do is you take a long strand of DNA, you elongate it on the surface, and once you deposit the DNA on the surface, you come in with enzymes, restriction enzymes, that recognize the local sequence of DNA and cut the DNA at precise locations. Once you have the cuts of the DNA, you can come in with robots that actually read where you have the cuts and tell you, back calculate what is the sequence, or give you an optical map of the DNA. Now this method works well, but one of the bottlenecks towards its full implementation is that most of the time the DNA molecules on the surface are overlapping with each other. You break them, you make turns. So when we started this project, we wanted to use theory and computation to design methods that would allow us to stretch the DNA on a surface without making any mistakes. And to do that, the idea was to rely on hydrodynamic interactions. So this is described over here. Let's assume that we have a DNA molecule in solution. It is a charged molecule, and we apply voltage. Because you have charges on the molecule, when you apply voltage, it will move from left to right. And as if it moves in solution, it is going to create a disturbance of the solvent far away from the molecule. And that solvent disturbance is going to come back and influence the structure of the molecule, which now, as it moves, changes the flow field again. So you have a highly coupled environment where the structure of DNA is coupled to the flow field, and the flow field is coupled to the structure of the molecule. That's what we refer to, these effects over here, as hydrodynamic interactions. Now what we'd like to do in our work is somehow take a glob of DNA that you take from a cell lysate and apply fields, voltage, and flows in such a way that you present the molecules in a manner that's amenable for optical mapping. So try to give you a pictorial idea of what these hydrodynamic interactions are. Let me show you a movie of a calculation that we did 6 or 7 years ago, in which we're taking a long molecule of DNA, about 200 microns in this case, and we're passing it through a small pore. When the molecule enters the pore, we apply a voltage that pushes it from left to right. This is an example of a simple Brownian dynamics simulation. It's a trivial calculation, nothing too interesting happens here. What is really interesting is what happens to all of the solvent when this molecule is being translocated. And that's shown on the lower panel. Right? So now the motion of DNA creates all of these flow fields. And once you see this, you realize that the interesting physics, what governs this process, is here, not here. And it is this coupling that we pay attention to in our research and that we're trying to understand better. Now, another interesting thing was that when we first did this calculation years ago, this took about a year and a half of computer time. And today, the same calculation using better methods and better computers takes only about 4 hours. So this gives you an idea of how rapid the pace of change has been. And many of these improvements have come not only from computers, but from better algorithms. So it is a very exciting field of research to consider for the future, because things will change, and they will change very rapidly. All right. So using these methods, one of the early designs that we came up with is illustrated here, right? So on the basis of a lot of calculations, we determined that the optical configuration, we would have these long channels over here, and then superimposed on these long channels, we would have nanoscale channels at a diagonal with a very carefully controlled angle over here, and then we would apply voltage in this direction. So instead of showing you the calculations, I'll just show you now the movie of the experiment, where you see the globs of DNA, the big channel, the small channel, and you see that as soon as the molecules find the entrance to the nanochannels, they enter and they become completely elongated. So if you stop the voltage at any point in time, you can present the molecules in just the manner that you want it. We're very excited at the time, and then we started to try to use these for devices. It became apparent that this was not reliable either. If you look closely to this image, you'll see that some sections of the molecules, for example here, are brighter than others. Here is another one. And when you have that, you don't know if what is happening here is that you have two molecules on top of each other, or perhaps you have an excess of fluorescent dye, or perhaps the molecule is folded onto itself. But you cannot use this for quantitative analysis in genome sequencing. So that pretty much put an end to the project. Another problem with this system that we had developed was that the stretch of the DNA, even though it looks like it is 100%, it was not. It was about 60%. That's the best that we could do with these hydrodynamic interactions. All right? And the loading was inefficient. So it was then that a very bright student came along and started to inspect, years later, these pictures once again, and realized that there were some cases where some of the molecules were actually fully stretched when you have a dumbbell here and a dumbbell over here. All right, so we call these giant dumbbells. And she thought, why is that? Why is it that when that happens, the molecule is really stretched out? So she went back to the models and started to try to manipulate hydrodynamic interactions in a way that would allow us to reproduce those configurations reliably. All right? This is a description of the models that we use. The details are not important. I'll simply mention that at this level of description, we describe hundreds of segments of DNA by a single interaction site. And then we have springs that connect the interaction sites whose parameters come from independent experiments. All right. So when you take that model and you use it to predict the motion of DNA molecules, you can calculate, for example, diffusion coefficients as a function of the molecular weight. You find that when you include hydrodynamic interactions, so those are the open circles, you have very good agreement with experimental data. Those are the diamonds. In the absence of hydrodynamic interactions, you get the incorrect scaling of these effects. Now, on the right panel, you see the predictions of this model when we put it in confinement. All right? So the question here was, can we use data that comes from bulk experiments to predict what happens when you are in a nanoscale channel? So there's two interesting things about this figure. We're plotting the diffusion coefficient of DNA in a nanoscale channel normalized by the value in the bulk, so by that number, as a function of the size of the molecule normalized by the characteristic length scale, the size of the confinement, the size of the pore. So two interesting things is that whenever the confinement reaches about 10 times the size of the molecule, you start to feel the effects of the confinement. The channel is 10 times bigger than the molecule, and you already have very strong effects on the mobility of the molecules. That's because hydrodynamic interactions are very long range. They're like electrostatic interactions. The second interesting feature was that a friend of ours at MIT did some experiments after the calculations were done to verify whether this was at all realistic or not. Those are the green symbols, and you see that it actually agrees very nicely with the models, telling us that we can go ahead and use these data to predict what happens when you go to very small feature sizes. All right? So using that, we started to try to understand how these hydrodynamic interactions work. We did some experiments, for example, trying to see how hydrodynamic forces would push the DNA away from a surface. That's what you see in these experiments. We have an oscillatory flow. As you go to equilibrium, the DNA gets pushed away from the walls and concentrates in the middle. You can measure this depletion layer and compare that to the predictions, and that's what you see over here. So these are depletion layers, these are simulations, these are experiments, and you see that the agreement is reasonable. We can use this for engineering purposes. And this is when the stroke of brilliance of this student came up. She said, why don't we use this effect to design devices that allow us to stretch these dumbbells using hydrodynamic flows? And that was one of the designs that she came up with. So you take a DNA molecule like this one, and you put it in a device where you're stretching it in this manner. The equilibrium state is for the entire molecule to be all here or all here. This is a non-equilibrium state. But when you are in this non-equilibrium state, these two globes over here and over here are pulling the molecule back and forth for a long time, for about a minute. And when you're doing that, you're creating a flow that is exactly like the one that I showed you in the previous movie, where you're going back and forth. So if that is what is happening, then this flow of this object going back and forth is actually pushing the globe away from the surface. When you push it away from the surface, you stretch the DNA molecule. And you stretch it here, and you stretch it here. Those are the physics. All right? So you can go ahead then and measure the stretch of the molecule, in this case as a function of the concentration of salt in our devices. And the interesting thing that you see is that when you take into account these hydrodynamic effects, the stretch of the molecules is close to 100%, which is much better than what we had before. It's the optimal device. In some cases, the forces are so strong that you start to unfold the DNA. You go beyond 100%. And the other thing that is very nice is that this agrees with experiments. So let me just show you again, as before, a movie of the experiment. You're going to see one of these giant dumbbells now in one of these devices. You see that the thing is going back and forth. Eventually, you go to equilibrium, and the entire molecule goes to one side. But when it is in here, which is about a minute, it is fully stretched, and you can analyze it. So the new devices now that we make, we've revived this project, are actually shown here. We have the dumbbells. Instead of breaking the molecules, we break only one of the strands, and then we add a fluorescent probe to the molecule. By using different colors for different sequences, we can know precisely what is the chemistry here, or here, or here, or here. And because we have 100% stretch, we have a very high resolution. So this actually works. The technology has been licensed to a company. They sell this. And they claim that with this technology, you can map an entire genome in 1 hour. Everybody tells me that this is not true, that it takes about a week. But that's not my problem. This is a company that makes whatever claims they want. Right? Now, on our hands, not in their hands, but on our hands, these devices continue to have some problems. What you see here is one of the channels that we made. And in these movies, you see that sometimes the molecules are not completely stretched. They actually fluctuate a lot. They go back and forth. This is in real time. They form the square patterns that you see over here. And they absorb to different surfaces. And we think that this is due to polarization and electrostatic effects. And to describe these effects, we need to come up with a model that has a higher resolution than what I was showing you earlier. So to do that, we started to review the literature. And what became apparent was that you have very good models over here. I showed you examples of what you can do with those. You have reasonable models over here, atomistic models. These actually do not work very well, but we have some available. But really at the length scales where you want to be working to build devices, you need to come up with something different and something that describes salt effects, mechanical properties, and the assembly of DNA. So the model that we came up with is shown here. It's called the 3SPN model, 3 sites per nucleotide. We have one site for the base, one for the phosphate, one for the sugar. We have electrostatic interactions. We have models with explicit salts or without them. It depends on your needs. What is interesting is how this model was parameterized. So we do rely on electronic structure calculations for structural features of the molecule, but that's about it. Everything else is actually coming from data, and the data include structures from the protein data bank, melting points, enthalpies, etc. And what enables the parameterization of the model very, very quickly is the development of new, very sophisticated sampling methods that allow you to do the fitting of all of these data very quickly. So these are models that now are kind of alive. Every year, lots of thousands of entries are provided by the PDB. We push a button, and the model is entirely reparameterized, and a new version is actually published. So you're always up to date with the latest data that's actually coming out. The new release will actually be this month. The last one was in 2013. All right, so let me show you a movie just to show you how the model actually works. These are 2 strands of DNA, complementary strands that are actually hybridizing with themselves. You see that you have this complicated dance where the DNA starts to approach. Eventually you make a key contact somewhere, and once you make this key contact, this is your transition state, you hybridize the entire molecule. All right? So one of the things that I said a moment ago is that coming up with methods that allow you to do this sampling very effectively is essential. One of the methods that we use is what we call the flux-tempered metadynamics. This is going to be critical because we need to be able to calculate melting points for this DNA to parameterize it. You see, a melting point is a very well-defined concept when you have a bulk material. When you have just 2 strands of DNA, we don't know what a melting point really is. So the way we define it is as the point where 2 local minima of your free energy landscape become equal to each other. We saw a very nice introduction to free energy landscapes. We calculate them in our work at different temperatures, and we find what are the conditions where these 2 minima are completely equivalent to each other. So in here you see a projection of one of these free energy landscapes. It has 3 local minima, dehybridized, hybridized incorrectly with a mistake, and hybridized correctly. You'll see a movie now that shows you what states we're visiting as hybridization proceeds. So you start over here, dehybridize. Eventually you make a key contact, but it's the wrong contact. You become dehybridized once again, and you hybridize in the right way, and you go to equilibrium. All right? So now we know what is a pathway, and we know what is a temperature at which this happens. So using these concepts then, you can calculate melting points compared to experiment. So these would be simulated melting temperatures for many, many different molecules. Each point is a different sequence. These are experimental melting temperatures. And you see that in general, the agreement is reasonable. The temperatures are correct, and then the width of the transition, that is the fluctuations that accompany this transition, are also correct. And you also see that these predictions are correct at different concentrations of salt. So the model really does what we had set out to achieve, and given this agreement, I think we can start using it to make predictions about what might happen in these systems. All right? So one of the things that one would like to calculate, of course, is kinetics of these reactions. So again, for the kinetics, you use sophisticated sampling methods. In this case, we use something called the forward flux sampling, which basically divides space into a series of very small transition states and follows the reaction along all of these transition states. All right? So what you get at the end of these calculations is a probability as a function of a reaction coordinate of the two hybridized, of the two strands being pseudo-hybridized here. All right? Let me show you a little bit how this works. All right? This is the probability that two strands of DNA are coming closer and closer and closer into contact with each other. You see that these curves have a maximum, a minimum, and a maximum. No matter what the sequence of DNA is, you always have this characteristic structure. Now this is very important because the reaction constant for the hybridization is going to be the product of all of the points that you see along this curve. Once you see this curve, you realize that this minimum here, the depth of this minimum, is going to actually control the numerical value of the reaction constant. All of the kinetics of hybridization is actually embedded into this point that you see over here. And this point is that mistake that I was telling you about a moment ago. That is the two strands coming to a close approach. They make a few contacts. Almost always, those contacts are incorrect. You need to wait a long time before you break those contacts and you form the correct ones, and the reaction proceeds. All right? Depending on the sequence, this minimum will be deeper or higher. All right, so we go ahead and we get reaction constants. And of course, we'd like to compare them to an experiment. See, before this work, it was thought that the reaction constants for hybridization do not depend on sequence. However, when you go ahead and use these models to look at poly-A or poly-AC or heterosequence, you see that the reaction constants actually vary by many orders of magnitude. So it's a very strong effect. Experimental data are available for a few sequences here, short sequences, short oligomers. They seem to be consistent with the calculations. There are no data, strangely enough, for poly-A or poly-AC. All right? So that remains to be validated. There are data for very long strands of DNA, and this is interesting. So these are polymers of DNA now. These are reaction constants as a function of molecular weight. The data start with molecules of 150 segments, so much bigger than that we have over here. These are all experimental points. There are no predictions. And you see that the slope of this curve is actually 1/2. When you look at our predictions, which seem to agree with the experiment, there's no slope. So again, this is another open question. Several things could be happening. Maybe the mechanism for hybridization is different here than here. Or maybe we do have a slope of 1/2, but we just cannot see it over here. Who knows? But clearly understanding how you go from here to here is critical and an open question. All right? Now for our own work, this is really what matters right now. In engineering, we use small oligomers to design composite materials. In the work of Chad Merkin, for example, he designs these beautiful nanocrystals that rely on the hybridization of small DNA oligomers. This is a growing field of research with more and more companies producing nanocrystals based on these hybridization mechanisms. These are some pictures of the crystals that one can create. You have access to all sorts of morphologies. And one of the weaknesses of these materials are their mechanical characteristics, for which we do not have experimental data. So one of the things that these models are actually being used for is to understand first of all, what crystals you form depending on the sequence of DNA, the strengths of interaction, and then of course the mechanical properties of these assemblies once you form different crystalline characteristics. And what is remarkable is that you can control the mechanical properties, the Young's moduli of these nanocrystals, by about 3 orders of magnitude by simply playing with the coverage of the DNA and the sequence of these DNA oligomers. And again, this is something that's coming out of these models really, because the measurements have not been performed yet. All right, so let me come back to this issue of chromatin then, and then show you what we're doing. So everything that I've talked about happened over here, just one strand of DNA. We talked about sequencing. We talked about materials design using hybridization. But what I told you at the beginning is that our end goal is to really try to understand how you go from that to that. This is one picture from one book. This is a picture from another book, very different. The point is we don't understand these images. Most of our efforts right now are trying to understand the histone complex and then the so-called 30 nanometer fiber. That's the name of this thing that you see over here. All right, so let's begin by discussing what happens here. This disc that you see over here is called the histone protein, and DNA wraps around twice. And it turns out that there's some really interesting elements of molecular recognition in these complexes. What you see over here is work by the late John Whittam, who actually realized that the binding of DNA to these proteins is actually encoded in the sequence of DNA. What he said was, or what he discovered, was that segments of DNA that have a very, very strong affinity for the histone have these TA motifs separated by 10 bases. All right? So a sequence like this one, right, it looks like a random sequence with TA motifs separated by 10 letters, has a very high affinity. A sequence like this one that has only one TA motif and nothing else will have a very low affinity for the histone. All right? So what would happen then in that case is that the histone with high affinity is stuck in position. The histone with a low affinity is moving all over the place and allowing the DNA to be accessible for transcription. That's very important in biology, of course. Now, the experimental facts are clear. Affinity is governed by TA positioning. The origin of this behavior is not understood. There's 3 hypotheses. The first one says that this has to do with the bendability or the curvature of DNA. So Bustamante and Spakovitz did AFM experiments. They deposited DNA on a surface, and using AFM, they were able to measure the local curvature of DNA. And they were able to come up with correlations that relate the sequence of DNA to the level of curvature that you see in these images. That work is heavily criticized, mostly because, first of all, the resolution of the experiment is not very high. And then what you're doing here is putting DNA on a surface and assuming that the on a surface behaves the same as DNA in the bulk, and that's certainly not the case. But that's the bending hypothesis. There's another one from a group at Columbia University that says it's all electrostatic interactions. They say whenever you have one of these TA motifs, they have a really large electronegative charge that binds to the surface of the histone. The DNA recognizes the histone surface. The more of these negative sides that you have, the higher the binding affinity. That's another hypothesis. And then there's a third one that is actually coming not by chemists or physicists, but by statisticians who looked at large sets of data, looking at the structure of DNA from the protein data bank, and they came up with a correlation that relates the binding affinity, the free energy of binding, to the curvature of DNA. Curvature of DNA defined in some manner that they propose in their work. And for a long time, this has been widely dismissed because they're statisticians, and we don't trust them we trust chemists and we trust physicists, but not statisticians. So we wanted to see if our model could actually explain really the origin of these behaviors. So the first task then was to come up with an equivalent model of the protein equivalent to the resolution that we have in our DNA model. So we have that. We take it from work by Peter Wallines and co-workers who've been working on these types of models. We bind this with the DNA and we start simulating the histone complex. All right. So in here you see these two terms of DNA, 1 and 2. We have 180 bases here, and then this is the histone protein, which has these tails hanging out that you see over here. All right. Now, if you think about it, we're taking two really, really different models. We're not adding absolutely any adjustable parameters. And we're hoping that somehow the marriage of that with that is going to work. So we need to validate this marriage of models. And we do that by looking at these experiments by Bustamante, once again, on nucleosome pooling. So he takes DNA with just one of these proteins, uses optical tweezers, he pulls, and he measures the force as a function of extension. And what he finds is that you have two transitions. One at about 3 piconewtons over here, and a second one at about 8 piconewtons over here. And he claims that these two transitions correspond to the unfolding of the first turn and then the second turn. That's why we have two peaks. He also finds or speculates that these transitions are reversible. You can go back and forth. So we do exactly the same experiment on the computer. We apply a force and we see how the system behaves. We measure free energy as a function of extension for different amounts of force applied to the molecule. Now what is interesting here is that, again, this is a non-equilibrium simulation. And what is interesting is that the minima of your free energy are going to remain the same, but the shape of the landscape is going to change when you apply these non-conservative forces. So let's look at just two of these curves. If you don't apply any force, so this would be this blue curve over here, the blue curve, you see that the minimum of the free energy, the equilibrium state, is a very low extension. That is, the DNA is fully wrapped around the histone. Right? The distance is short over here. If, on the other hand, you apply a really large force, you would expect this to become unraveled, and the equilibrium would be a very large extension. And that's what you find. If we look at this curve, which is this one, the equilibrium state, the minimum of free energy is a fully extended DNA. It is that case. And in between, you have barriers. So now the question becomes, how do we calculate the pseudo-equilibrium state for this non-equilibrium process? Well, you do that by analyzing the heights of all of these barriers and when they cross each other. And when you do that, you can construct a curve that looks like these are the barrier heights as a function of tension. You have dotted lines and solid lines and dotted lines. When these lines cross each other, you have the equilibrium force for the transition. And you find that the two equilibrium points are actually 3 piconewtons and 8 piconewtons, which, if you think about it, is remarkable. These are two very different models. The experiments were telling us 3 and between 8 and 9, and this is giving you only that. And the reason this works so well is that, it turns out that this is governed by very, very strong forces. Elasticity of DNA, which we understand really well, and electrostatics, which we understand really well. And the details do not matter at all. They're completely overwhelmed by these types of forces. So there's no wonder that this actually works. And that's the reason biology works. If biology responded to all of these tiny other forces, it would be a mess. Right? It has to be a robust system that doesn't make mistakes. All right. So now that we've validated the model, we can actually start making predictions. One interesting, for example, is what happens when you start messing with the histone tails that you see over here. It is widely believed, for example, that modifications or the loss of these histone tails is actually associated with diseases or with aging, with getting old. So when you go ahead and repeat the same calculation with tails or without tails, you find that the actual affinity of the same sequence of DNA for the protein is much, much weaker. It becomes really easy to unravel that, and it becomes very easy for the DNA to zip around the histone. You can calculate what happens for specific modifications, and you find that some of them are actually very, very sensitive to the force, some are not. So these, I think, will provide clues as to how some of these aging-related diseases are coming up. By the way, this paper came out today, so this is brand new research. You're the first ones to see these things. All right? So now we go back to the binding of DNA to the histone. All right? So we can calculate what is the probability that a given sequence of DNA binds to a histone. This is the free energy, so the log of this quantity is the probability. These are predictions of the model. These are experimental data. And I'd like you to focus on just two curves, the green one and the black one over here. In the green one, we've taken a model of DNA that has all of the forces that I was telling you about, but absolutely no curvature. That is, the DNA is straight. And you see that the predictions are really, really bad. In the black one, we actually have the curvature and the electrostatic forces. If we turn off electrostatic forces, you get the red points, which are actually worse. What this is telling you is that the affinity of DNA for the histones is actually completely governed by the curvature. So these statisticians, the Italians, they were actually bang on the money. They were correct. You see, we are educated to think that DNA is a rigid rod. All models of DNA except ours are like that. The DNA, depending on sequence, has a solid curvature. And this curvature, how much of it you have, determines how tightly you bind to histones. So that's where it encodes genetic information. It's a purely geometric consideration, which again is an incredible surprise. All right. So I'm running out of time. I was going to show you now how you move around in DNA and how you go to fibers, right? So I'll just leave you with a little taste of what the next hierarchy of models do. Depending on the local sequence of DNA that you have, you will form strands of DNA that are really stretched out, and then these 30 nanometers fibers that you see over here, where you have different structures in coexistence with each other. And again, this is completely governed by the sequence of the strands that you have over here. Now, this is just the 30 nanometer fiber, but these are the first predictions that are telling us that the local structure of these large objects is going to be different depending on the chemistry of the molecule. The next step, of course, will be to go to thousands of these proteins, and of course to put this in a model of the cell, which we're doing now. I won't be able to show you that, but if you ask me a question, I can show you a very nice movie. Thank you very much. We will take some questions. Please. Laura.
Okay, so my question is, I see in the dynamics of your DNA simulations that the curvature of the DNA is almost fixed, right? Am I correct, or?
No, the curvature of the DNA.
The small curvature of the DNA. I mean, so, because the question is, have you tried to simulate, instead of DNA, for example, RNA that has loops? And the loops are, I mean, the function of RNA is mostly in the loops that form the RNA, right?
Yeah.
So now that you already have all the parameters for the DNA, maybe you can try and try to predict. I mean, the RNA loop prediction is a very important subject because
Yep.
that's the function. so you can try to simulate RNA with your...
So you're absolutely right. It's very important. Yes, we have tried, and that no, it doesn't work. The results are really, really bad, and the reason is that in this model we only have 3 sites. RNA is actually a lot more complicated because, as you said, it can fold onto itself. It can hybridize with itself. And to describe that process, you need a model that has more sites than just 3. You need at least 6. So we're developing a new model to do that, but it's going to take us many years. So,
And then I have another question. So I see all the parameters of your DNA and your simulations, but you didn't mention any of the simulation process. Did you build the machine to make the simulations, or you use a different software like
So we have put all the parameters for this model on LAMPS, which is a public code. On August 30th, so this month, we will release a new piece of software that aims to replace LAMPS. It's called SAGES. It's based on GPUs. It is a lot faster, and all of these models are available there. And you can download everything for free from our website. Yeah, so there's a group at Oxford that has a model of DNA that can be used to design origami. It cannot be used for anything having to do with biology because it doesn't have charges. The hybridization is incorrect. It doesn't have curvature. The mechanical constants are not. But for origami, it is a very, very good model. It's very fast. And probably for origami, it's better than ours because it's cheaper. So yeah. Well, you want to go into origami? No, I think we are not.
When you talk about this double dumbbell, and then you talk about the efficiency of stretching,
yeah.
Then you showed a graph of the stretching as function of the ionic strength. Do you have I don't know, a microscopic insight or how the concentration of ions affect the stretch of DNA?
Yes. So first of all, you stretch the DNA because when the concentration of salt is very, very low, the charges on the DNA molecule are no longer shielded. So charges along the phosphates of the DNA repel each other and cause the molecule to stretch. All right? What is more interesting is once the molecule is stretched, the flow patterns in the channels change. And you can go from an electrosmotic process to an electrokinetic process and change the direction of the flow. And those physics we do not understand. That's another. Yes.
Regarding different ion concentrations, does the model capture B to A to C DNA transitions, for example?
Yeah, it does. For DNA, it's very good. If you move away from DNA like RNA, it's not good.
Another question?
Yes, sir. About the electrostatic interaction, these are modeling with Nernst-Planck equations or some kind of models. What about, for example, if I have a lot of salts, it will interact with the DNA, and the migration of the species charged may interact with the DNA and broke the structures. So what is the limit of the electric field applied, and what is the accurate of the measurements?
Yeah, so depending on the problem that we're trying to look at, we use simple biomarker, or we include an explicit description of all the ions in the system, so full electrostatics. Our model doesn't break, so it's, those physics are not captured by the model. But solving the Nernst-Planck equation at the same time as we solve the hydrodynamics becomes really important when you go to very, very salt concentrations, where again, you transition from being electrokinetic to electrosmotic flows. And that's a key part of the problem when you work over here in this regime.
Thank you, sir.
No more questions? Okay, we will thank again Juan de Pablo.
Thank you.
We will have a small break, a short break. We will resume in exactly 10 minutes.
¿Ya la descargaron, no? ¿Son estas? No sé cuál sea la de ella. Empieza Julia. Es que aquí ya descargaron 3, pero no sé. Esta es la... No, esta es hasta el rato. Julia, ¿está en un directorio?
Sí, está en México, porque había traído la película por si no
No,
funcionaba.
esa no es.
Con
Esa
México
es la
con
segunda.
ahí.
Ya.
Sí, el PowerPoint, por favor. Ahí.
¿Y cuál de las
abajito.
dos?
Un PDF, sí. Vine preparada por si no funcionaba.
Ok. Vamos a ver eso. ¿Lo ponga?
Sí, en el bolsillo.
Sí. Sí, se queda, yo ahorita le bajo el volumen ahí arriba para que no, ya se...
¿Cómo a qué altura? ¿Por aquí? ¿Tan cerca?
Pongo, ahorita pongo
Ok.
el ponente pasado lo tienes acá.
Ah, vale.
¿Me escucha? ¿Sí?
¿Sí? Sí, encantada. Sí. ¿No hay para pasar las diapositivas? Sí, porque yo soy de pasear. Yo creo que Andrés tenía, quizás se lo puedo pedir. Ah, vale.
Porque la batería estaba...
Ah, no, pero para pasarlas.
Ah, no tengo idea.
¿Tú tienes? Ah, perfecto, gracias. No, lo que quiero es poder pasearme yo también. Pero, ¿cómo funciona? Carlos, ¿cómo se conecta? How do you connect it? Ah, vale. Está escondido.
Sí.
Sí. A ver. Ahí va. Ahí sí, sí, ya está.
Okay.
¿Sí?
Well, welcome again. We are now going to begin our second part of the one section. The two talks we have now are in the field of what we know as quantum mechanical topology, where we analyze scalar and vector fields of relevant molecular functions to understand structure and recognition. So we have our first speaker, Julia Contreras, who is a very prominent and active researcher in the field. For example, we know her from her contribution in the development of the NCI index used to characterize intermolecular interactions. Now she's going to talk us about kinetic energy density. So please welcome our speaker now.
Thank you. Thanks, Jesus. And that's too loud, right? No? That's okay? Okay. So I would, first of all, I cannot say I did exams here, but I totally know how it feels like, and I didn't know how it felt like having wonderful workshops back in my university. So you're very lucky. Make a profit from it. And I certainly feel very happy to be back to Mexico. I came here 2 years ago. I fell in love with the country, so this time I decided to come even 10 days before. So I'll show you a little picture at the end. So as Jesus said, we work on NCI, which is based on the reduced density gradient. And what I will be talking about today, it's also on the reduced density gradient and a generalization on how it works based on kinetic energy densities. I'll also talk about the electron localization function. I'll introduce what quantum topology is. And I'll try to show you why we care about that. So try not to go too far ahead from my talk, I'll start with a motivation. Basically, why do we care about the chemical bond, or rather, why do we still care about the chemical bond, which is not an easy question to answer when you start working on that, and how we care about it, which is indeed quantum topology. And I'll introduce three functions: the electron density through the atoms in molecules theory, the electron localization function for the localization of electron pairs. As you see, I always move a lot to stop me from falling. And the reduced density gradient for the analysis of non-covalent interactions. I think that one of the important aspects of developing these theories is not only being able to explain all approaches, such as the Lewis theory or the valence shell electron pair repulsion theory, but we also want to go beyond that and understand new chemistries. And for that, I will show you a couple of examples on high pressure where we have new electronic structures, and we need to understand where their properties are coming from. From there, I will merge into a more physical description on how these functions work, the kinetic energy density, as I was saying, how to understand the reduced density gradient features, how they can take us, they can actually bring us also results similar to the electron localization function, and even intramolecular bonds that we could not see with atoms in molecules. And since we can look at, I'll show you covalent bonds, non-covalent bonds, intramolecular bonds from the same background, then I'll show you this. It's a perfect tool to understand reactivity. So that will be my last point, and then I'll just dip into the summary. So starting with the motivation, to myself and to my students, I basically try to convey the fact that nowadays we have very complicated methods, and we can have very nice quantitative predictions. So basically the computer can do the work, but I would like to understand it too, and that's a sentence from Wigner. And since I want to understand, and what I want to understand is chemistry, let's go back to Pauline's definition of chemistry, which is the science of substances, their structure, properties, and the reactions that change them into other substances. If we try to analyze this sentence, then we have structure and reactivity as the background for them having properties. And if we want to understand that from the microscopic point of view, then we have to understand where are the chemical bonds and how do they change into one another. So we basically need to really understand what's going on with chemical bonds. That's the main body of our work. And to do that, we have, basically that's what I was saying, why do we still care about the chemical bond? It's a very old concept. It's 100 years from Lewis' paper on electron pairing. And that's not an easy question to answer sometimes. I got it asked by my father when I defended my thesis. And believe me, he could not believe we were still caring about the chemical bond. And the thing is that basically the chemical bond was born before the advent of quantum mechanics. It's a very nice concept to generalize behavior of substances, but it's not quantitative nor predictive in many cases. If we want to go quantitative and predictive, we have to go to quantum chemistry. And now our wave function is delocalized. So we've lost all these nice features that enabled us to classify and understand. So we want to keep the best features of both worlds, and for that we have to build a bridge. We can build that bridge in many ways, and one of them is looking at the dimensionality. When the chemical bond was introduced, people were thinking in 3D. That's basically the way we first study the chemical bond. We have balls and sticks. And 3D is also a quantity, a dimensionality where we know we can start digging into quantum chemical results from the Hohenberg-Kohn theorem. So basically what we do in most cases in quantum topology is we use this dimensionality. We go to 3D to understand what's going on. And I'll try to explain you what we do in quantum topology in a very simple manner. Basically, we have our wave function, we calculate our wave function, and then we use a chemical function. I always work with the scalars, but you can also use vector fields. So basically, you have, oops, shit, okay. Okay, that always happens. Okay, okay, and when that happens, it never comes alone. So, where is the laser? Vale. Sí. Okay, so basically we have a chemical function, and I'll tell you more about that in a minute, and we calculate it from our wave function in the whole space. Computationally, that basically means we calculate it on a grid. So we have a function, and what we do is we just analyze it like you did in your math courses. So we analyze the shape of that function. We analyze the maxima, the minima, the saddle points. We can also go for topological concepts, which is basically we divide our system into regions like we have here into, that would be associating a region to every maximum of the system, and then we can integrate those, and we can have, since we're talking about a chemical system, if we integrate the electron density, we will have the charge associated to its region or the volume. And since these regions have a chemical meaning, let's say that this region is a bond, then we can have the volume or the electron or the charge associated to that bond. Obviously, the key point here comes in there. So what chemical function are we going to use? The first function that was analyzed topologically was the electron density in what is known as the quantum theory of atoms in molecules introduced by Bader. And that's basically how it works. Here we have the electron density of benzene across the plane. The electron density has a cusp, or what we can associate to a maximum, in every atom in the system. You have all the carbons around here and the hydrogen atoms. So then we can recover these gray spheres for the carbon and white spheres for the hydrogens. Well, what he did is, now let's say that we can associate the saddle points in between maxima, so these points, to chemical bonds. And if we do that, we obtain the red spheres in here. Now you say, okay, that looks pretty obvious from the electron density, but that actually enables us to draw those lines that we really had a hard time to guess when we were building up our chemical intuition in the first years of chemistry. So we end up with the chemical structure of benzene, with carbon-carbon bonds, carbon-hydrogen bonds. So for example, we do not get saddle points here, so we don't have hydrogen-hydrogen bonds. So that's a way to use electron density topology, it's the shape of this function, to have, from ab initio calculations, chemical structures. In this approach, these points, the bonds are associated to points. But as I was saying, if we want to have things like bond charges, which are absolutely necessary to understand chemistry, we need to have regions associated to those bonds. So then we have to use another chemical function, and that's the electron localization function. The electron localization function, I will build on it later on with a formula, was introduced by Dickey and Edscombe in 1990. It is close to 1 in basically where you expect a Lewis pair to appear. So that here we have the nitrogen molecule, and you have the lone pairs, the nitrogen core, the nitrogen-nitrogen bond, then again the core, then again the lone pair. So we have a function that can recover the Lewis picture from ab initio calculations. As I was studying that as an undergrad, I found this wonderful. And that's a 3D picture for a molecular solid. You can, oops, again, okay. See, Mine works the other way around. I'm going to keep doing that the whole time. I can see that. Okay, so now, for example, we have the lone pairs here. As you can see, they are bigger than the bond. That's valence shell electron pair repulsion theory we are recovering here. Another important thing about ELF is that it is 0.5 for the homogeneous electron gas. So not only we can have a look at localized pictures, but also at delocalized ones. Here we have the picture of a metal. That's sodium. We have that's a map of ELF across the solid. And we have the cations with the shell structure. This one is a little bit lower. That's why we don't see it as well. We have the cation, and then you can see this all around it is pretty flat. That's the delocalized electrons in the metal. Now, as I was saying, that's very neat. We have recovered Lewis picture. We can, I have shown you in a very brief manner how we can also look at valence shell electron pair repulsion theory. But that's something we already knew. We didn't need quantum mechanics to do that. We want to go beyond that. And we want to go beyond that because we have now new experimental techniques that push our concepts. We are faced against new electronic structures, and we cannot understand that sometimes from the old principles. So as a theoretical chemist, one has to really update those definitions, understand what is the electronic structure and how it is related to the properties. And that's one example. Metals such as sodium or potassium, under very high pressures, they become optically transparent and insulating. So what I say to my students, they don't behave like metals anymore, right? Those are the main characteristics of a metal. So for sodium, that's like a 200 GPa, a 5-fold compression. It's a huge compression, but it's something that can be done. It's actually done compressing in between. It's called a diamond anvil cell, and you compress it, and it's currently done in many labs. As a matter of fact, that's also one of the neatest results in the last year and a half. They found a new superconductor totally different from the very complex phases they were analyzing, and that was just hydrogen sulfide. They just needed to put it under pressure, and the critic temperature went up to 100 Kelvin, which is a huge difference from what was being done. So we really need to understand what's going on with these new experiments and where is this coming from. So basically, what is the electronic structure behind those new behaviors? And going back to metals, that's what we had for sodium at normal pressure. Now we go to high pressure, and that's what we get. We still have the cations, and now you see this is very low ELF. We don't have the delocalized electrons anymore. Instead, we have that. And those are extremely localized. They didn't have a shell structure. There's no core inside. Those are the electrons. The electrons are not delocalized along the sample anymore. They have collapsed into localized bodies in the voids of the structure. So that's why we call them pseudo-anions. So basically, that's a pseudo-ionic structure. We have cations, and we have pseudo-anions. The only thing, they don't have a core inside. So that's why it doesn't behave like a metal anymore. It behaves like an ionic structure. So as you can see, this picture is extremely easy to obtain from calculations, and it gives you a strong insight on what's going on. It really makes you understand, to a first-year chemistry level, the properties of the sample. Now, just so that you can see how deeply localized it is, those are the cations, spherical of course. Those are the localized electrons. If we integrate the electron density inside it, it makes like 1.8 electrons. And what's really funny about it is that we have seen that metals can become insulating, that localized electrons really behave like anions. And we can even go for new laws of understanding how these things are happening, because these pseudo-anions actually appear in the same symmetric positions, same Vikov positions as real anions in well-known structures. So this is an example for potassium and nickel. Obviously, they appear at different volume, but what this is really telling us is that there is an equivalency between external, so hydrostatic pressure and chemical pressure that can really help us understand how these materials are going to behave. So we have seen atoms in molecules, we have seen the electron localization function, but the thing is, the big question is this really all? Here we have two pictures, and as instead of just one benzene like we had before in atoms in molecules, now we have a stacking of benzenes. This stacking obviously does not happen through the localization of electrons. So nothing appears for ELF. When we look at the quantum theory of atoms in molecules, we have two bond critical points, so two first order cell points. So basically the stacking is seen as a two-body interaction, a carbon-to-carbon interaction twice over here. But stacking is related to dispersion. And dispersion should be a benzene-to-benzene interaction. There is a sum over all atoms in the C6 terms. So what we really wanted to do is to go beyond that localized picture of chemical bonding, especially for weak interactions, and try to understand better those non-covalent interactions. And for that, we used the reduced density gradient. The reduced density gradient, which is this function, very well known in DFT, basically we have a constant, the modulus of the density gradient and the density to the 4/3. We can use that to understand non-covalent interactions because it gives us a very differential behavior whether we have interactions or not. That's the way it works. So we have a model density for a hydrogen atom, and now we plot, if we analyze its behavior, we can see that the reduced density gradient behaves like rho to the minus 1/3. So if we plot the reduced density gradient in terms of the density, we have this behavior, and we want to look at non-covalent interactions, so that's low density. If we go to low density, then the reduced density gradient explodes to infinity. Now we have two densities, and even if we do a promolecular approach to just a sum, that's what we obtain. We see that in between them, because there is a bond critical point, but not only because, the reduced density gradient will go into 0. So now if we plot two hydrogen atoms at a long distance, then we see that this goes to 0, okay? The reduced density gradient goes to 0 in between them, and if we do the same plot as we had before, that's what we obtain. So we have these two curves. The first one, it's this rho to the minus 1/3 behavior from the outskirts, but we have a new part, and this new part is related to the part in between the molecules, the atoms, whatever you have in there. So you see, we have an extremely differential behavior. This is going to infinity, this is going to 0, and that's going to happen every time we have an interaction. So we have an easy and very clear way of identifying interactions, weak interactions for the moment. We have two curves in here, red and green, which is the self-consistent and the promolecular approach. So just the sum of the densities or the relaxed densities. As you can see, they're very close to one another. So that's the reason why this method, I won't be showing it today, but it can be used to very big systems. We have used it for proteins and DNA within the promolecular approach. Now let's go to a real system. Here we have formic acid. That's the calculated density, and that's our rho to the minus 1/3 behavior. Now we put two of those together. We have the dimer. They're interacting, and as you can see, now we have peaks. Those peaks are related to the interactions in between the monomers. Now what we do is, okay, we have a way to know that there are at least two interactions there, but we also want to visualize it like in the ELF approach. And for that, we just choose a value of the reduced density gradient cutting those peaks, and we plot an isosurface of the reduced density gradient, and that's what we get. So we have here a picture of the hydrogen bonds like the ones that Andres showed before, and a little dispersion over here. And that's because we can also differentiate different interaction types. We can go from a strong and attractive, like hydrogen bonds, to weak, very weak, like van der Waals, or even strong and repulsive, like a steric clashes. So we have a whole mapping of the different types of non-covalent interactions. Here you have some examples. We have water dimer appearing in blue. We have methane dimer, obviously in green, and a strong steric repulsion in bicycle octane. So 3 textbook examples where we can see where the non-covalent interaction is. We can also differentiate in different molecules where we have mixed interactions, hydrogen bonds and van der Waals. The stacking in benzene, you can see now that it really is a benzene to benzene interaction, delocalized interaction, or even the 3 of them, we can go to big systems like Andres already showed. Something very interesting is that we can also use the shape to understand the natures. As you can see here, we have a circle shape that's localized interactions. And as you have more of a sheet, then it means that many atoms are contributing to that interaction. So we have seen one example where we can use the electron localization function to understand new chemistries. Now let's see how we can use also non-covalent interactions to understand new chemistries. And that's an example where we had some experimental colleagues coming to us because they had this arsenalide compound, which is formed by AS406 gauges. And it is, when you compress it in most pressure transmitting media, even alone, it amorphizes at around 15 GPa. Now you do that in helium, which is supposed to be inert and very innocent transmitting media, and that's what happens. It actually goes into the voids of the structure. So you see here the helium atoms inside, and you might think, okay, maybe it's just, helium is innocent, it's not interacting much. Well, that's not what it's actually happening. Here you have the picture, an amplification. You have helium here, and remember how the hydrogen bonded looks like. Here we have localized and with a strong density interactions with the arsenium close to it. And even if you might think, okay, so what does this mean? Well, that means that actually when you have this sample compressed in helium, the amorphization does not happen anymore. The structure is stabilized, and that basically made our colleagues really afraid because many, many experiments have been done on helium, and now they have to check whether every time they had voids in their structure, that was being, the results were being affected by the pressure transmitting media. So that's something that theoretical chemistry and experiments can really work together and understand even the conditions, the best conditions for an experiment. Now, now that hopefully I have convinced you that it's nice to look at the chemical bond, we wanted to understand better the physical meaning of the reduced density gradient and even of the electron localization function, etc., how they were working, why are they revealing so well the chemical structure. And it all starts with kinetic energy density. So now we start with the second part. It all starts with the kinetic energy density. If we can picture the bond between two atoms with localized orbitals, you can see that these maxima are going to give us a low kinetic energy density. So electrons are slower in these regions. And that's what goes into the formula of ELF, which we're going to reviewing here. So we have the kinetic energy density. But the ELF wants to reveal the Pauli principle. So it's not only about kinetic energy density. So we have the kinetic energy density, and what we do is we imagine a bosonic system with the same density. And that means that we put all the electrons in the same orbital, and that's the associated kinetic, the bosonic kinetic energy density. And that's why this is called the polykinetic energy density. Now you say, okay, so that should work. That should give us the Lewis picture, and we should have minima every time we have localized electrons. Well, that's the dirty washing from ELF. That does not happen. Okay, that's something I already said. That does not happen. That's what the polykinetic energy density looks like. So basically, it's maximal at the nuclei, but we don't see anything. That's nitrogen molecule. We don't see anything for the nitrogen bond. And that's because the kinetic energy density has an electron density dependence. So it's overridden by the electron density at the core. So what Vicky and Edscom did, they just divided by the main dependence on the density, which is the homogeneous electron gas. And when we do that, we get rid of the density dependence. That's the picture we get. So now we have a minimum, so the electrons are slower in the core, in the bonds, and in the lone pairs. Now we're used to thinking rather in orbitals. So they just did a mapping so that everything is reversed. And they did a Lorentzian mapping so that we have the typical ELF picture. Now we have lone pairs, core, bond, everything down again. Okay? So that's the typical way of looking at the electron localization function. That's the 3D picture, again with the lone pairs and the bonds. Now, we wanted to understand all these terms really and what was going into the formula. So we started looking at it in another way, very simple way. Now, we tried to understand, okay, so what's coming from the kinetic energy density, so from the normalized kinetic energy density, and from the bosonic contribution. And that's something that was already known. That's the localized orbital locator, which also Vicky introduced. And that's a new term which we call the bosonic kinetic energy density, which is also normalized. Now, if we look at how it behaves in the nitrogen molecule, obviously, the bosonic contribution is a lower bound to the localized orbital locator. That was not me this time. For once, it was not me. So, that was just to wake you all from when you saw the formula. Okay, we have the localized orbital locator. Obviously, the bosonic contribution is a lower bound because the electrons are going to move even faster because they're fermions. When electrons are in what we call completely localized orbitals, to a very good extent, which happens in the core, you can see that they basically coincide. They do not in the valence, where we can see the effects, rather the effects of their fermionic nature. Okay? So basically, here we have very high densities. They spread rather to the valence. Now, this means that, which is pretty intuitive, if electrons are coupled, they really behave like bosons. That's one of the principles also behind ELF. And so that means that if we look at the bosonic term over here, we can also see the shell structure. And that's what we have in here. So if we look at T-bows, we have, that's the image for nitrogen, we have the covalent bond, we have the shell structure, we have the lone pairs. We can even look at what people call charge-shift bonds. That's a very similar image to what ELF gives. Charge-shift bonds, it's usually understood as resonance between ionic terms, and that's the ELF picture of it. So we have this term, and now the funny thing is that very simple math shows us that it is related to the reduced density gradient. So we have that the bosonic term is 5/3, 5/3, the square part of the reduced density gradient. So NCI, we saw that it identifies non-covalent interactions, and now we see that it should also identify shell structure. And that's really what happens. That's basically merging the two features together. We can look at the electron localization features, so at the shell structure, lone pairs, bonds, because electrons behave like bosons in those regions. We can also look at non-covalent interactions. And in order to understand how to merge these features in topology, we look, as I told you, we look at the shape of the function. So what we do is, okay, now let's look at the shape of this function and see when do we expect these features to appear. So where are the critical points? So we look at the gradient of the bosonic term, the normalized bosonic term, and that's what we obtain, which basically means that we have two types of critical points. We have the first type, which we call atoms in molecule critical points, because it appears when the density gradient goes to 0. So that means that when we look at NCI, we're recovering bond critical points, ring critical points, gauge critical points. Since we're looking at isosurfaces, we recover regions around those points that I showed you in benzene, for example. And it can also go to 0 when this other term goes to 0, and that's the second type of NCI critical points. And when this term goes to 0, that's related to the optimized electron potential. We have shells, lone pairs, like we did see right before. We also have another type of, another way of making this go to 0, which is very interesting because it's related to intramolecular interactions, which are very difficult to capture from atoms in molecules. And those are closed shells interactions where the density gradient does not go to 0. The density gradient does not go down to 0. And those are closed shell because it's very easy to show that the Laplacian is positive in all these points. Now let's see at one example. Let's look at one example. Those are the diol series. So we have butane diol, propane diol, ethane diol. And the experimental evidence shows us that the OH stretch vibration is going from strong to moderate to weak. Now if we look at the electron density, that's what we have. In the first two cases, we have a bond critical point. So everything matches nicely with the benzene picture I showed you at the beginning. We have a hydrogen bond in between these OHs. Now the problem is that there is no critical point for ethane diol. So according to atoms in molecules, there is not even a bond over there. But the map, the electron map, looks pretty similar, right? So so actually for these three, what we have is that we can see how the density here is bigger than in here. So the interaction is becoming weaker, but there is no data for ethane diol. There is no bond according to that theory. Now if we look at NCI, which is somehow generalizing that concept, we have in the first case, remember I showed you, when we have a localized interaction, it's more kind of a circle. So we have a strong interaction, very blue. Now it's becoming more delocalized, and at the end there is no critical point. Those are the little points over there. We have no critical point, but we still have a delocalized interaction according to NCI. So we can match the experiment, and we can even try to understand, go back to the theory I just showed you, and see how this is actually coming from an atoms in molecule critical point. The second one is not. It's coming from the second part of the equation, where this goes to 0 and the Laplacian is positive. And that's very, very easy to identify if you're looking at the 2D plot like this. In the first case, the reduced density gradient achieves its absolute minimum. It goes down to 0. It does not in the second case. So these things are extremely easy to identify if you're doing this calculation, which is actually extremely fast. And then you can also go and analyze these minima and the values at these minima, so you can see how the electron density is going smaller all along the series. So we've seen that With NCI, that's something that's where we started. We can look at low-density regions. We can even look at intramolecular bonds, which are very weak. But we can also have a picture of localized electrons and shell structure. Since we have shell structure, covalent bonds, non-covalent interactions, even ionic interactions, which I haven't shown directly in here, but you'll see them in a minute. Now, obviously, let's look at the reactivity, which is another huge part in chemistry. And that's one example we chose because along this reaction, we have covalent bonds, we have polar bonds, we will have ionic bonds. So, and obviously we have van der Waals and non-covalent interactions. So as you can see, the hydrogen is moving, and this interaction is getting stronger as the hydrogen is being transferred to the NH2 part. You see it's getting bluer, so this hydrogen is going to be transferred. And now, at the transition state, you see everything becomes extremely flat. So basically, we have, that's where atoms are easily transferred. And now we have the new bonds being formed. This hydrogen really leaves towards the NH2 group, and now we have the other hydrogen going towards the lithium, which now becomes a new unit. So that's extremely easy to calculate, and it gives you a really picture, it gives you a very intuitive picture of chemistry and reactivity. Another way to use this, we have seen this example where we have different bonding types, but we can also try to use it in cases where we have more subtle chemistries and where we actually need non-covalent interactions to understand what's going on. And that's one of the examples we did was with chiral recognition, because usually we have the complex with the main interactions that determine the stability of the complex, but the chiral recognition happens through another weaker interaction. And that's a way to look at it. We have here the example we worked on with some collaborators. We have the complex, the homochiral complex, where we have the OH-NH interaction, and it has similar main binding interactions as a heterochiral. But this one is favored, and it was postulated that it was due to a T-shaped interaction because it was like a 2 kcal difference. Now, T-shaped interactions are also very easy to see with NCI. That's the way it looks. They basically have this cone shape in between what is interacting with the pi system. Now, if we look at our complexes, that's what we obtain. We have the main interactions in both cases, OH, NH, OH, NH. And now we also have this, that's the CH pi interaction here that should favor the heterochiral complex. So we have from strong bonds to weaker bonds. And, okay, so I'll jump this part. Unless you can ask me afterwards. Now, everything is being implemented also for MD to understand ensemble densities. So I'll just go down to the summary. So what I really want to transmit here, and I'm sure Carlo will go deeper on that, is that topology can really help us in the description of new chemistries. We have seen 2 examples, insulating alkali. We have seen strong helium bonds. As far as high pressure is concerned, and if you like films, that's the way I sit. That's Straw Dogs, Perros de Paja. We have a math teacher going to his wife's village. He really goes with the stereotype of a math teacher. He's not violent. He's pretty much staying on his own. Well, after very high pressure from the medium, you can see he pretty much changed his face. So everything has a breaking point, and topology and chemical bonding theories in general can help us understand those. We have seen basically that non-covalent interactions have a signature in the, that can be calculated from the electron density and its gradient. It can go from van der Waals interactions to hydrogen bonds to steric repulsion. It's applicable to small and large systems. I haven't shown examples, but we saw some in the morning. We have organic, inorganic, solids, etc. We have seen the meaning of NCI, which is revealing regions of bosonic behavior. It reveals lone pair shells, cell structure. It recovers atoms in molecule critical points, but also non-atoms in molecules critical points, which is very useful in intramolecular interactions. We have seen that it can be used for interactions where different interactions where we have covalent and non-covalent, but also subtle reactivities. And just a quick, I'm very happy now NCI is implemented in several packages. The ones we work on actively are NCI plot and critic. Carlo is working in NCI Milano, and those are the people who helped me through this work. Roberto, Fernando, Tatiana, and Christophe. Collaborators both in high pressure, solids, experiment theory. It's really nice to work with all of them. And that's the picture I have. I really needed to show you. That's, I came here 10 days before, and I went to Holbox, and I was able to swim with this fish, which is the biggest fish on Earth, el tiburón ballena, so the shark whale, they call it. It's not a shark, and it's not a whale. The poor thing, it's just the biggest fish on Earth, but apparently that's not enough. They needed to give it a new name. And that's something we see in many cases in science, and we certainly see that in many cases in chemical bonds. Apparently things need bigger names than their concepts. That's
Thank you for the wonderful and very provocative talk. I mean, one question many questions, there are many questions. But first, and perhaps the most naive, why did you use the positive defined kinetic energy density? How do the picture change if you use the non-positive kinetic energy density? I don't know. I mean,
that's a very good question, and that's something we haven't tried yet, though we have thought about it. In topological approaches, historically, they chose the positively defined one because it's easier to interpret. If you think about it, if you want to get rid of the bosonic contribution, which is positive, then it wouldn't make any sense if it could go negative. It wouldn't be a lower bound. So I guess it all started building on from there, but that's something we really, I mean, this is pretty new. This is from this year, so.
Thank you. Two short questions. One is the expression, you used the expression bosonic region. Now for me, bosons are totally different elementary particles than electrons. Yeah. Do you have region of bosons there, or why do you use this expression?
We use this expression because the kinetic energy density term, the bosonic kinetic energy density term is extracted from having all the electrons in just one orbital. So that's what the image of, that's the image we want to convey. And it really, I mean, we started analyzing it all from ELF. And
yes, but there are still electrons there, not other
elementary
particles.
Totally.
So I find
but
the expression a bit
trying,
unfortunate.
when you use this term, you're trying to annihilate this spin that makes them keep occupying higher orbitals.
Okay.
That's,
Second short question, you said you have strong helium bonds. How strong are they?
That's something we haven't looked at yet. And it's not that easy, because I guess you could go for an energetic partition, but in solids you have so many interactions. I see it as I don't think I even would like to do that.
Well, we found helium bonds 3 kcal years ago, so I would be interested how much you found there.
But since, it's not like a small molecule where you can do small bits and pieces, because you have all these gauges and you have so many, especially when you have the helium in and things are compact. You have so many long-range interactions. I don't know if I'd be happy with an energetic partition.
Why do you call them strong when you do not know how strong they are?
Well, I can certainly count on the electron density and the fact that the solid does not amorphize anymore. That to me is the experimental evidence. Because they do in all the other pressure transmitting media, and they tried like 3 or 4, even without, it does amorphize. And when helium is there, it does not anymore.
Okay. Thank you for your nice presentation. Actually, I got a kind of confusion, because recently I've read a paper that they also studied a couple of molecules, and they find out that in some molecules when we do QTM analysis, and it might be possible that we do not see, critical points, bond critical points. But if we take a look at the delocalization indices that we obtain from AMOL, then they will show, significant delocalization and bonding. Yeah. If in such cases, what's the difference between taking a look on the delocalization indices when we do not see the critical points to speak about the bond, weak bondings, and the use of NCI that you presented here?
So
Thank you very much.
It's going through different ways, but it's basically giving the same answer. So if you want to look at it from the shape, from the topology, these cases are related basically to what is called in topology very low persistence. That means that when you have huge mountains and valleys, you can use the maxima to identify what's your general picture. But let's say that you're already in the mountain and you have just like little peaks over there. Then looking at those maxima is not going to convey the real picture of the mountains you're looking at. You'd rather look at it as a whole. So that's what we're doing with the radiosensity gradient, and that's if you actually look at bond critical points. Carlo and Angel have a very good paper on that. They are related to exchange. Now, the way I see it is if you have kind of a huge surface of interaction, then that's going to give you also a greater delocalization. But that's something that we should formalize.
So we have time for some more questions.
Okay. Mine? Thank you, Julia, for a very nice lecture. I have a question concerning the arsenolite. When helium is inserted, there is a change of volume and expansion or not? And if there is a change of volume, could be this, the lack of amorphization should be just due to this, or there is real bonding?
The change in volume is negligible, because they use the voids of the structure. They use the voids when it goes inside. Obviously, when you keep compressing, then it doesn't behave the same way anymore.
Okay. Thank you.
I just have a quick question. Maybe people have done it, maybe not, but when I see papers on this field, and people always have this visual interpretation that this looks like a cone and this looks like whatever. Has somebody used machine learning or some automated tool so that you just give it a molecule that identifies all these features rather than a human having to rotate this thing in 3D and say it's a cone or not? Has there been research on that? So that you just run it into a machine and you get some...
Yeah, totally. Those are not very common in the community, but there are ways, global and punctual ways. The easiest one is to look at eigenvalues that will convey the shape of your interaction very easily, so just like in any functional analysis. Well, those are okay, but obviously it would be better to have a global way, like you won't have, it's easier, how to say. So with the eigenvalues, it's very easy to tell whether you have a localized or delocalized interaction. But it's not that easy to have, for example, a T-shape, which to the human eye is very easy. So then you need a global approach. And we've been talking to informaticians, specialists who are specialists in the topological approach, and in their fields they already have, obviously, tools to identify that. So that's something we're working on.
We have to change here things. I don't know. ¿Qué tal? ¿No? Vamos a cambiar solo la presentación, pero espérenos aquí porque no vaya siendo. A ver, pues ven.
¿Cómo?
Does it work?
¿Es mejor here?
You have to put it on your focus, on your belt, and el cinturón.
Oh, here? It's okay?
So which is your, maybe this one?
Yes.
This one?
Yes.
Sí, creo que sí. ¿Me puedes cerrar un poquito? Escape. Okay.
Animaciones. And
you can use this one.
That's the principle. Okay, I think it's okay.
Okay, ready?
Go.
Okay. Well, specialist in the field with very important work in chemistry in terms of electron densities, and of course the developer of the source function very useful descriptor for structure and reactivity in the solid state conversion of quantum chemical topology. So let's see what he has to tell us today. Thanks a lot.
Okay. Thank you. Buenos días, buenas tardes to all of you. And I wish to congratulate the Facultad of Química and with the Institute of Química for their so important anniversaries. And I wish to thank the organizing committee for this prestigious invitation and Jesus for the introduction. Okay. Well, I'm essentially a theoretical computational chemist, but my talk today will be focused on what comes after modeling. At the very end, one has to compare and validate the result of her modeling against some experimental outcome, which in my case is often a crystal structure and possibly a charge density derived from X-ray diffraction experiment. And if such a comparison has to be meaningful, it must be as much as possible unbiased. That is based on analyses that are fully independent on the method that I've been using to get either the experimental or the theoretical outcome. And if in this comparison I use descriptors that are related in a way to chemistry, then as a side result of the comparison, I will get also some precious chemical insight. Okay, I will use descriptor based mainly on electron distributions, say electron density, electron spin density, being defined in terms of quantum observable and measurable quantity, like the electron density to a very accurate X-ray diffraction experiment, then they allow a direct and as much as possible unbiased comparison of theory with experiment. On top of this, they may provide description of chemical paradigms which, at least in principle, is freed from the very beginning from any model preconception or arbitrariness, though arbitrary may be the way in which I'm using and processing the observable. And they are also ideally suited to validate or possibly reject those interpretive models of chemistry which are based on useful but still arbitrary objects like molecular orbitals. So the question that I want to address today is whether subtle but challenging chemical effect may be still detected through simple electron density descriptors. Okay, I'll say a few issues about current problems, chemical bonding studies, especially in thin crystals. Well, first, detection. Is really the bond path, the bond critical point, the only available criteria or usable criteria to detect them? And once I have detected, how I classify them? Well, it will depend on the bond descriptor I will be using, and these will be related to the source of information on bonding I have, either only the electron density or the first order density matrix or the pair density. And, density. Okay. And the source of information will be related to the kind of experiment I have performed or the level of initial theory I have been using. And clearly, all these aspects are very much interrelated. Well, with coherent elastic bracket scattering, I have information on only the diagonal part of the first order density matrix, that is electron density, which contain only indirect information about electron correlation, but on the other hand, it allows us to obtain important descriptors like the Laplacian, the electrostatic potential, electric field, the reduced density gradient that have been introduced by Julia, and the source function which I will be talking today. And out of a diagonal element are instead available from for at least in principle from inelastic scattering, counter scattering, and as the normal result of an ab initio computation, and from the first order density matrix, I can derive the energy densities that Julia has been talking about, the ERF as well, and for instance, from a singular terminal approach, I can also get this, the second order density matrix, so the pair density. From the non-classical part of the pair density, I may derive the whole family of very nice, very important descriptor of chemical bonding, or the family of the electron localizability indicator, domain average Fermi hole, and in particular the localization index and localization index, which allow me to us to get the number of electrons that are fully localized on a basin, also number of electron pairs that are shared between any pair of basins. Okay, which are the, I think that the current trend, the chemical bonding studies in crystal may be now described in terms of two parameters. One is the level of contamination between the experimental and theoretical outcome, the degree of their independency, which in my view contamination must be as low as possible. And then the more or less relevant weight that information I take from experiment has on the conclusion which I get on a bonding situation. In my view, this relevant information from experiment must be as high as possible. Whereas now it is customary to have studies where a lot of independent information is taken from the scriptome using the first order and second order density matrix, so requiring a wave function, while the weight of the information coming from the X-ray is very marginal. So the obvious question is why to perform an experiment in this case? Another approach is a constraint wave function method due to Dylan Jayati-Lakar, in which a wave function for a system in vacuum and placed in a periodic box is modified in order to get theoretical structure factors as close as possible to those from experiment. Well, at the end, one gets a wave function that feeds the crystal field, but assigns the problem of the arbitrary weight lambda to be assigned to the constraint of maximum likelihood between the theoretical structure factor and the X-ray experimental structure factor, one gets a wave function which is no longer variational. And so in this case, there is this problem, and the contamination between experiment and theory is very large, and the amount of information from experiment just depends on the Lagrange multiplier related to the constraint of maximum likelihood I mentioned before. Very elegant is the method of trying to reconstruct the first order density matrix from several kinds of elastic and elastic scattering experiment, and try to fit the data from this experiment to the parameter of a model wave function of first order density matrix. This method is in its infancy, is seldom feasible, but it is very promising, and clearly the contamination between experiment and theory is small, and the level of relevant information from experiment is very large. Well, my current philosophy is simple, is to go beyond, in a way, the charge density topology, but still using descriptors that use only information of the electron density. So either the reduced density gradient NCI that Julia has mentioned for non-covalent interaction, or the source function, which has proved quite efficient to describe non-trivial bonding situation. And clearly in this case, the contamination may be even zero because all this can be obtained from experiment, and so this is very large. Okay, source function. Well, this function has been introduced by Richard Bell and myself a long time ago. In a nutshell, it enables to see the properties of electron density at a point of the space in terms of source contribution from all the remaining points of the system within an interesting cause-effect relationship. While in practice, the value of the density at a point is related to the behavior again of the density, but in terms of this Laplacian, at all other points of the space R prime. And clearly one gets a tool which is perfectly tied to one of the main operative rules of chemistry, that any local properties in chemical behavior is to some extent, be small or large, influenced by the remaining part of the system. And in mathematical terms, well, the electron density at R can be seen as caused by a local source, which has this expression. The part encircled in pink is just a Green's function, or the influence function, which represents how effective is the cause, the Laplace of the electron density, in contributing to determine the effects of electron density at R. Then I can replace the operation of the local source over the whole space in separate operation over the disjoint exhaustive partitioning given by the zero flux recipe of the Bader theory. And so in this way I can express the electron density as a sum of atomic contribution, each of which is called the source function from the atom omega to the electron density at R. And clearly this function is amenable to experimental determination because one just needs to know the Laplacian of the electron density. And well, with this decomposition of the density, the properties of the density may be viewed from a totally new perspective in a way. Well, the density is no longer a local properties, whereas they originate from the cumulative result of the influence of all other parts of the system. In a way, this is nothing new. In the FT language, we would say the electron density is a unique function of the external potential, that is the position and charge of all the nuclei of the system. But with the source function, we may investigate the local response of the electron density to a perturbation, internal or external, in terms of contribution from all atomic group or atom of the molecule, how they do contribute in giving this local response. So we are using the language of chemistry. So in a very simple example here, this is a structural series of metal formamidinate complexes, just replacing the palladium with molybdenum and looking at the reconstruction of the source function, of the density at the bond critical point between the two metals. In this case, from a bond order 0, we get a spread of source with dominance of the ligand contribution. This must be multiplied by 4, whereas here the sources are much more localized and are determined substantially only by the 2 meters. Okay, and does this decomposition provide a real chemical side, or is it just mathematical identity? So, and well, let's start with a very simple example, covalent bond between carbon and carbon. We want to reconstruct the density at the bond critical point. We look at the percentage of sources with respect to the total density I have to reconstruct. And well, the volume of these balls is proportional to the percentage source function. Well, we need is a covalent bond. We expect that the major contribution must come from the 2 bonded atoms to which the bond critical point refers. And this is that so, and this increasing bond order. Let's see a less trivial case, diborane, and we look to the BH bridge bond. And well, in this case, the source function for the 2 bonded atom is much less than here, and it is asymmetric, denoting the polar nature of the bond. And so much spread sources. When we go to the terminal BH bond, again we get a value that is very close to that normal covalent bond, but asymmetric because it's a polar one. So I will present now some example concerning hydrogen bond. I have no time to discuss this, which will be interesting. How to use a source function to go beyond the bond path criterion. How can be used to test the concept of hypervalency. Whether the source function may detect also electron localization effects. And then extension to electron spin density. Okay. Let's consider how the source function describes the change of the nature of the hydrogen bond when the donor to acceptor distance changes. And from doing this, we take as a Gedanken experiment the approach of 2 water molecules along the water dimer linear CS constraint. And so we are exploring distances that range from those of the weak electrostatic in nature hydrogen bond down to that of the charge-assisted hydrogen bond. Okay? Sorry. And we immediately see that the source function from the hydrogen involved in the hydrogen bond to the hydrogen bond critical point is an incredible marker of the nature of the hydrogen bond. While it is largely negative for weak hydrogen bond, and then it becomes increasingly larger and eventually positive in these cases. Well, another thing, if we look to the source function contribution from the hydrogen in both the hydrogen bond and the acceptor oxygen, we see that it never reaches the value typical of a covalent bond. We need to add at least the donor oxygen to get a value of sources close to the covalent bond, which means that the source function tells us that the hydrogen bond is at least 3-centered in nature. And this has been, for instance, shown recently also by Popelier using delocalization indices, but this is done without using the pair density and was shown much time before. And well, when one could be surprised by having a negative source. What does it mean to an atom that contributes a negative electron density at that point? Well, if an atom is in isolation, it can certainly produce only positive source because it is alone in producing that density. But if for a polyatomic system, it may happen that when an atom is strongly perturbed in its shape and with Laplacian distribution, which is very far from spherical symmetry, then this can happen and has a profound chemical meaning. For instance, here we see the change in the shape of the hydrogen going from this situation to this situation. When the distance from the hydrogen to the receptor donor molecule become closer, much equal, and so we have a charge-assisted hydrogen bond, then this becomes much more symmetric, the distribution also, and so since here we have region of positive Laplacian close to the point where I'm constructing the density, so negative local source contribution, which are not counteracted by the positive charge source contribution given by this region, but when the nature changes, then I get positive value. So there is a profound physical meaning in the negative sources. And when I take real hydrogen bond, then I find the same effect. I'm not discussing them because they are only a bit exaggerated with respect to the model I was using before, but they are the same. What is interesting is that if I look to the resonance-assisted hydrogen bond, then I observe an increased delocalization of sources. The sum of these sources relative to the distance that I'm using. This is comparable to that, despite this bond is 2.2 angstrom longer. And this is an effect of the resonance-assisted hydrogen bond. Okay. The source function enables us to, in a way, to solve an apparent inconsistency of the QTM theory. I refer to the concept of bond path. Clearly, it's topologically associated to the only two atoms it connects, so it is linked to a two-center vision of the chemical bond, whereas we know that chemical bond may be multi-center in nature, may be fractional, they may be competing scheme of bonding. On the other hand, we know that the bond path shape and all the values of density along the bond path clearly depend on the whole set of interaction. But using the source function, this apparent inconsistency of QTM clearly fades away because any point along the bond path, and in particular the bond critical point taken as the most representative point of the bond, of the bonding interaction, depend not only from the two linked atoms, but in principle also from contribution from the whole set of other atoms in the molecule. So, which brings to the fore non-local effect on bonding. And well, the source function does not even require a bond path to detect an interaction. This has been shown very nicely by Farrugia when he studied this iron trimethylene methane complex. Well, it's an experimental and theoretical study. He found that only one bound path is observed between the iron and the carbon in alpha, and no bond path with the carbon in beta. But then when this is close to a structural catastrophe point, so just with slightly changing this angle, then appears three more bond paths with between iron and the carbon in beta. So this is a eta 1 or eta 4 iron complex. Well, if we look to the delocalization index, we even found a larger delocalization between iron and carbon beta than with carbon in alpha. So, and also other, say, the NMR barrier to the rotation of the trimethylene methane indicates that there is an interaction between all the ligands with the ion. And indeed, when we take the source function description, we see either we take this reference point or this reference point, we see similar spread of sources. This atom is contributing to here more than that, and this again, and there is no real change when a small changes in geometry is done. So the source function gives a description that is very close to that provided by the localization index, despite the bond path is selected. Okay, let's see the source function to test the concept of hypervalency. Well, as all we know, the octet rule is one of the cornerstones of chemical bond theory, introduced by Lewis in 1916, but then challenged by Linus Pauling to explain bonding for third row elements. Well, he supposed that the sulfur has to expand its octet using the orbital, giving partial double bonding, and so hypervalency. And the sulfate anion, this picture seems to be supported by short SO bond length. Well, one can write also a structure for the sulfate in terms of only single bond, but this is at odds with the electron neutrality requirement of minimizing the formal charges. So hypervalent sulfur, single bond picture, we thought it important to make a study using a charge density, so completely freed from any orbital model. And we did so using combining a very accurate charge density analysis with initial computation on the periodic crystal. And we use a crystal of potassium sulfate where the strong ionic environment should ensure that the sulfate anion has a charge very close to the formal one of -2. Well, note that in this case, the normal tools that are used to distinguish between single double bond cannot come to a conclusive decision here. If we think to the single picture, so sp3 hybridization, the oxygen, and so we can expect three lone pair, three non-bonded maxima, whereas we see a double bond picture, sp2 hybridization, two non-bonded maxima. Well, it cannot work, because in both kinds of structure, all the oxygen atoms should reflect either the static or resonance occurrence of three lone pair. And actually, either experimental and theoretical, we found three non-bonded maxima. We can use also the concept of, or elasticity that is used normally to decide about partial double bonding, pi bonding character in planar conjugated system in terms of the elasticity, that is a departure from perfect cylindrical symmetry of the distribution of the density along the bond. But in our case, this is not useful, because the sulfate anion has almost TD symmetry in the crystal, so the elasticity will be zero regardless, almost zero, regardless of the sign, single double nature of the bond. So both these criteria cannot be used here. Well, let's take the source function, and we immediately reconstruct the density at the bond critical point of SO. We immediately see that the oxygen gives twice the contribution than the sulfur, so a quite polar situation. And quite interestingly, the other oxygen are surprisingly giving a contribution that is almost as big as that given by the sulfur. If we look to the delocalization index, well, we see that localization index for SO bond does not exceed 1. And there is a partial sharing between the oxygen, confirming what we have seen with the source function. Let's do an experiment. Let's decrease the distance from equilibrium, maintaining the TD symmetry of the sulfate anion, while we would expect an increase of covalency and some some partial double bonding and a decreased charge transfer. This is not so. We see that the bond is becoming more polar. This is increased, this decreased. We see that the charge transfer is increased, and we observe anyway something that seems not related to this, because there is charge that is transferred from the non-bonding region of the lone pair in the bonding region. This decreases sensibly. But this does not serve to increase the covalence of the SO bond, but just to increase the sharing between the oxygen in a resonant way. As we see here, this increases, and also this increases. So in a way, the source function is able to solve the condition of a decreased lone pair population without increasing the covalent and double bond character of the SO bond, because it enhances the delocalization among the oxygen. And in a way, we find something that is reminiscent of the old Center for Electron Bond model of Pimentel and Randall, where placing electrons in non-bonding C2 orbital just gives an explanation of the resonance, the sharing we find between the oxygen. This sharing is favored if we shorten the SO bond, but this does not lead to a double bond nature, but maintain a single SO bond because the sulfur is excluded from this process. Okay, so let's discuss now this. Could you use the source function to detect delocalization, electron delocalization effect? And if it works, can we apply also to X-ray derived electron densities? Okay, suppose that we know that there is electron delocalization between atom B and A. Does this have any effect in the way the electron density in B is determined by A and vice versa? Well, we have addressed this in these two papers. And let's start first saying that clearly the best way to reveal electron delocalization effect is to use the pair density, which, as we define, is that we have just talking about. And in terms of these delocalization, localization indices, I can immediately see, for instance, that in case of the benzene, 44% of electrons are fully localized on each carbon, and 22% of the electrons are shared with the neighboring carbon in ortho, 2% carbon in meta, 4% carbon in para. So we get a very sound picture of pi-electron delocalization. So why using also the source function? Well, the answer is does not require the pair density. So I can apply both to experiment and theory. A disadvantage, it seems to lack some direct physical connection to electron pairing, but as advantages in a way because it may provide quantities that tell us locally the influence of more or less distant remote atom or group of atoms, and differently from the DIs, lead to an atom bond rather than atom-atom connection, and then the full freedom I have in the selection of the reference point enable us to see how different part of an atom or group of atoms feel the effect of electron delocalization effect. So this can provide chemical insight, as we will see. Okay, but we have to look to the effect of electron delocalization. Well, it has been claimed, for instance, that source function will be unable to see this effect for a pi conjugated planar system because a null contribution from pi molecular orbitals to the electron density in the nodal plane. However, this was not the end of the story because sigma and pi distributions are not independent but are self-consistently interrelated. And so one can expect that some, albeit small effect on electron conjugation can be still visible when the reference point lies in the pi nodal plane, even though in this case clearly pi orbitals play only an indirect role. And here are the results. I consider this system with supposedly enhanced delocalization from here, from left to the right. And well, we wanted to reconstruct the electron density at this C1, C6 ECP. If the source function is able to detect electron delocalization effect, I am expecting that the contribution from the two bonded atoms should decrease on going from left to the right, whereas the contribution from the other atom has to increase as an enhanced electron delocalization effect. And this is actually what we find. And well, now let's allow the pi electron to enter into the play. What we do, we move the reference point above or below the bond critical point in the direction of the pi distribution. And we see that these effects, small effects, are very largely enhanced. Note that this capability to reflect pi electron delocalization is completely independent on any sigma pi separation. I could have obtained the same results using a electron density given on a grid, for instance from a maximum entropy method calculation experiment. But if I am using a molecular orbital model, then I can also see the separate contribution from sigma and pi MO, and actually I see that this increase in the nearest neighbor contribution going from the molecular plane up to 2 atomic units above or below the plane is essentially due to the pi part. So, well, can we extend all this to experiment? We took the electron density of 2 molecular crystals of 2 prototype homoaromatic molecules, benzene and naphthalene, for which very accurate data sets were available. And we also consider a bigger system with data of standard data quality and with the advantage of looking whether the source function patterns that we will find here are transferable to some extent to the benzyl and naphthyl moieties of this molecule. And clearly, if they are transferable despite different molecules, different data set, a different accuracy of the data set, this could be really important to look at it. And we can also investigate whether the source function is able to sensitive to the substituent effect. Let's start with the benzene crystal. We took the data from Burg et al. We did 2 multiple refinements, one including, the other excluding the hexadecapolon carbon. The first gives a better agreement, but assigns a too low elasticity to one of the carbon-carbon bonds. And here are the results where we compare for BA, NN, and OT, the experimental result with those from a periodic calculation of the crystal, and we compare also the result of the 2 multiple. Well, as we see, we find a same a very nice qualitative and quantitative agreement between all these data, despite we are looking to a very subtle properties like this electron delocalization effects. And standard deviation refers to the average of data over the asymmetric unit of benzene, which contains 3 bonds, half benzene molecule. And clearly these are larger for the system with hexadecapole because, and in particular, out of the molecular plane, because we had a bond whose ellipticity was badly described by this model. So everything seems to be Okay, so let's go to this system, and which has two phenyl groups, similarly substituted, and two naphthyl groups, differently substituted. Okay, let's compare first the two benzyl ring with the benzene crystal data. And we see, despite a different system, different data set and accuracy, we see a very nice agreement between all the source function pattern, and this also above and below the ring. Okay, the effect of substitution here can be seen only in the standard deviation, this value, because I am averaging over all the 6 carbon-carbon bonds in the phenyl ring, and this is decreased when I remove from the average the bond involving the carbon link to the bridge carbon, those yellow ones. Well, let's see what happens to naphthalene. Consider first naphthalene alone in vacuum, and clearly one bond becomes shorter than in benzene, the other one are longer, so the source function pattern are discriminated by these distances, as you see. And this ranking of ordering of source function percentage is found also when we take the crystal data for the theoretical, and the same ranking is also, ordering is also found when we take the data from the experimental density into the crystal. And the average value are due to the fact that the symmetry is reduced to see only one inversion center is present. Well, let's now look to the likelihood of, with naphthalene, for the least perturbed ring 1 of the imino-binaphthol system. And we see that for this least perturbed ring, again we find a similar ordering of sources. So transferability is ensured in this case. But if we look to the most perturbed ring, then we express the deviation of source function contribution relative to naphthalene, and we see that a clear deviation occur only for those bonds that have at least one or two carbon substituted. So in a way, the source function is immediately able to spot the substitution, spot transferability, but also the effect of substitution. I think I skipped this because it's more technical, and I give you some conclusion about this part. So I think the capability of the source function to reveal electronization effect is firmly established both for theory and experimental electron density. And since that the results are stable against different data sets and systems, so it allowed to talk about reproducibility and transferability. And the fact of chemical substitution is, however, clearly evident. I think that is really remarkable, the match I found between experimental and theoretical outcome using the same unbiased tool to detect subtle electronization effects. And I'm not discussing this, but I can tell you that if you use an independent atom model electron density, one does not recover the source function pattern trend that one get with either the real wave function or the multiple model experimental electron density. So these trends are not due to geometrical effect, but a real electronic effect. Okay, why extending the source function to the electron spin density? Well, we can give some insight on how spin information is transmitted from the paramagnetic to non-magnetic center, and whether these latter, in a way, are able to influence the spin density distribution of the paramagnetic center. Or more in general, whether an atom or group of atoms is helping or is counteracting the paramagnetic center in determining the spin polarization at a given point. And if so, whether it does so in a relevant or negligible measure. Okay, it is quite trivial, this tension, because I have just to replace the density with the spin, both in the expression of the local source, so the local cause here is no longer the Laplacian, but the Laplacian of the density, but the Laplacian of the spin. The effect is clearly the spin and not the density. And the green function remains the same because it is just a geometrical feature. And then the partition in atomic contribution, even in this case, is still done in terms of electron density because I want to refer to quantum atom of the theory and not, so this needs to be done so. Okay. Well, let's compare the source function reconstruction for water triplet at the bond critical point OH. As we see, they are completely different in the case of the source of the density or of the spin density. Why is that so? Well, we have changed the effect, so the cause is different, and this means that the density and the spin density dilute or concentrate themselves in a completely different way to the space. And this is why we have this, certainly. Dilution concentration is expressed in terms of the Laplacian, clearly. And this was recently published. Okay, now, well, if we look here, we see that while in practice the interpretation of source function contribution for the spin density are apparently much less easy than for the density. Here we are looking at the bond critical point and we have dominant contribution from the two bonded atoms and marginal contribution from the remaining hydrogen. Instead here we have similar contribution from the two hydrogen which also oppose the contribution from the oxygen. Why is that so? If we look to the non-bonded charge concentration relative to the lone pair in the molecular plane, if we look at the source function reconstruction for the density, it is completely independent from the method I am using. But if I use the source function of the spin density, there is a dramatic change according to the quality of the method. Why? And for a simple system, it is already difficult. It becomes intractable. It is a nightmare when you consider a more complicated system like this double bridged azide copper complexes. So what can we do? Well, first, let's consider the relationship between the local sources and the densities. In the case of the electron density, it's very simple. When the Laplacian is positive, that is, the electron density is diluted, the local source is negative, and the effect of the region around R prime will be to decrease the electron density at the point where I am reconstructing the density, and vice versa when it is concentrated. Well, for the spin density, the situation is the same, but if I wanted to understand it in terms of the composing alpha and beta quantity, then I see that regardless they are all diluted or all concentrated, the final effect will depend on the relative magnitude of this quantity. I call the effect alpha if if the region around R prime is contributing a positive spin density, beta if it is reducing the spin density, so giving a beta effect. And for instance here I see that if the two density are concentrated, then if it is the alpha density greater than the beta density in magnitude, then the Laplacian of the spin will be less than 0, the local source will be greater than 0, and I will have an alpha effect. Otherwise, I have a beta effect, despite these are both concentrated. Then it is important to decompose each local properties into what I call magnetic contribution from the unpaired alpha electron density, plus a contribution that is due to the remaining electron density. Okay. I call this reaction because it is a reaction to the presence of an unpaired alpha electron density, or relaxation because the alpha beta constraint is no operative here. And clearly this we integrate to the number of unpaired electron, whereas the spin component of this remaining electron density will integrate to 0, but locally will be differently from 0 in general. And how to single out these magnetic orbitals? Well, the first option is not feasible. I don't discuss it. Possibly one can take the single occupied orbital of the reciprocal open wave function. This provides an ambiguous decomposition, but for a model wave function which is very poor, one with spin density greater than 0 everywhere, and with a reaction contribution set to 0 everywhere. Much better is to take the natural orbital from a diagonalization, the first order density matrix of a wave function with some kind of correlation, say CAS-SCF, UHF, spin contamination annihilated wave function, and take those natural orbital close to 1. This is in general unambiguous. And I can show some results. Maybe I skip this, so even if it is interesting, we can see that the magnetic part, despite it is done only by alpha density, can, as region of negative Laplacian of the spin, so this region will produce a beta effect, despite it is all beta density. Okay, this is how we compare the spin density and electron density, and that for the spin density, a reconstruction at 4 points. And I see that in this case, in general, the magnetic part doesn't play a role, but only in this case where it is reconstructed the density related to the concentration of the impaired electrons. Whereas in this case, both play a larger role. But before trying to understand them, just introduce this notation. Green means alpha effect, red means beta effect, and the sign of the SFS is positive if the atomic source is concurring to the density, to the spin density reference point, or it is instead negative if it is opposite. Here is negative despite it is an alpha effect because the spin density at the bound critical point is negative. So it's against the effect of the oxygen. And just try to understand this. Well, this is very nicely understood because nothing, as I said, is contributed by this marginal, by this hydrogen, so it is a true bond effect. We are describing a bond interaction. Whereas in this case, there is first, the hydrogen are opposing to the oxygen, so there is a spin polarization mechanism which operates not only through bond, but also through space. Why? Because these two give a similar contribution. Why is that so? Because if we look at the magnetic relaxation part, the magnetic part is in a way not related to bonding, but is just related to spin delocalization, and this is, and this, that is more related to the bonding part, is comparable to that. So give this value. In this case, instead, this part magnetic is about 1/4 of this, whereas here that is related to bonding is marginal with respect to this. So compared to all this, one comes with this final results. I think this was explained here. And well, this is for lone pairs. We see that this is a CAS-SCF wave function and just show what happens when I introduce a static and dynamic correlation with respect when I do not have this correlation. Well, I immediately see that this magnetic part, no, this non-magnetic part the reaction part is very badly described by UHF. It is not even contained by ROHF. And despite, if I look at the absolute source function, I see that the magnetic part is already very well described by all the methods. It is the other one which is not described well, so in a way the source function enables also to get a very good insight in why and where a given low-quality method fails. And actually, one of the most important reasons is that CAS-CF increases the spin density by a factor of 5, and this is almost due to the non-magnetic part. Okay, I think this is a conclusion, and if we have no time, I just 1 minute. Okay. So, in a way, the spin density source function seems to be able to highlight how spin information propagate from paramagnetic to non-magnetic center, and the way that the paramagnetic center spin polarize the non-magnetic center, and or the extent that this latter back determines the spin distribution of the form strongly depend on the chosen reference point, as we do expect. And this is because of the large anisotropy of the spin, and even more so the spin Laplacian distribution. I think decomposing the magnetic and the reaction term is very important to get chemical insight. The first term does not necessarily determine a positive spin density at any reference point, but maybe also yield a beta effect. And the relaxation term either concur or counteracts the magnetic term determined S at RP. We saw that this simple wave function leads to trustable magnetic orbital density, but not to the reaction component. And clearly joint analysis of the source function for S and R provides interesting insights. And well, coming to the experiment, clearly the source function of spin density is amenable to experimental determination. One has to use polarized Newton diffraction data and X-ray together, where it has been recently shown by Lecomte group that one can get a good spin density for the experiment. And use the source function in this case would be of very large importance, because on a way we have shown, we know the large sensitivity of the spin density distribution to the adopted theoretical framework. And on the other hand, there are known technical limitations and ambiguity in the multiple modeling. So combining the two would be very interesting to improve both methods. And in the case of experimental spin density, so function atomic contribution, one has a problem of partitioning in magnetic reaction component. One has no the natural orbital machinery, but one could make use of the grid partitioning function, which is defined to the relative weight of the corresponding terms from theory. And well, thanks all mentioned plus photoquarters, and clearly all of you that have been here despite it was late, and we are all wish to go to have lunch, I think. Okay, thank you very much.
Is it there a systematic study to relate the source function to electron delocalization phenomena such as aromaticity, for example? I mean, you mentioned that you can use the source function for study electron delocalization. Has this been used for, you say, to study aromaticity in a set of compounds?
Well, I think that yes, when one of the papers I've shown, yes, I have used this for, I've defined also there was a JCP paper, And I use this also to define an index like the index based on Fermi Hall, the realization indices and so on. And it has a very nice agreement, say the correlation of about 1 with these other indicators, so it seems to work very well. No, I am saying, just trying to, and well, now today I stress more the part of the experiment because this was not anticipated, and I was glad to find this. You're welcome. Yes, thank you.
In the case of non-covalent interactions from experiment, okay, there is a problem, right, with the weak interactions, for example, from X-ray diffraction experiments that is carried in the source function because you are also including the Laplacian and this is sensitive, for example.
Yes.
What can you say about this, please?
Now this is a good question, certainly. Clearly the source function is dependent on the Laplacian, but the source function integrates the Laplacian over the Bayesian. So it is the result of an average. So why actually we are able to find good results is because we are taking an average. In a way, so locally the Laplacian, experimental Laplacian, theoretical Laplacian may differ a lot. But when you took the average, both they have to integrate to 0, as we know, on the Bayesian. And actually, the reason why it works is this one, which we are not looking. Locally, we look at the density, which because actually we are using the Laplacian to reconstruct the density that we know that is good. So in the average process, we are in practice avoiding all this kind of problem.
More questions? No? Well, let's thank Professor Hybridization for his talk. And also to all of our speakers in this morning session. Bien, hay un anuncio. Están listas las mamparas para quienes van a poner los carteles, pueden hacerlo desde ahora. Gracias y nos vemos, por favor, a las 3 de la tarde. Bien, Luis tiene la...